Hi Pankaj, are you there?
Hello.
Hi Pankaj, are you there?
So Pankaj sir will be there in a minute.
Okay, fine, fine. So I was just wondering that why the share content option is not active.
It's already 1 minute. I have not given the right actually.
Oh, okay. I see. So once you
Okay.
give the right, then it should be fine, right?
Yes, sir. I will give you right now. You can try.
No problem. No problem. Yeah.
Give me 2 minutes.
Okay, sure. Please take your time. I just wanted to make sure that things are fine.
Now you can say it. Please check it.
Oh yeah. Now I am able to see it. So which one is the best option to take?
I mean,
Which one is the best option to
you can
choose?
take you can take a screen or a window both. Both are same. No difference.
You mean screen one? That's the which is auto selected. That's the
Okay.
one.
The screen one will show only the screen which is you open, and window
Okay.
whatever you will do in between to get the skin. All thing will be shared.
Okay,
So
so Windows Explorer is the best option you are saying, right?
better will be screen share. Open your PDF and or PPT and share that screen.
Yeah, that I understand. So what I'm trying to say is that when you click on share, right?
Mhm.
So then it shows multiple option.
Mhm.
So okay, screen one is already selected. So and then other options are Google Chrome, Microsoft PowerPoint, Windows Explorer.
All the files are which are open in your window will be shown there. You have to select which one you want to share.
Yeah, I can share the entire window. I have no issues.
Okay, then it's fine. You can share it.
So which option to choose for? That's what.
As I am saying, if you share your window, everything you will do between will see we can see from here. But if you share your screen, only the screen will be shared here. So it
Yeah,
will
I
share
understand. Okay.
screen. Right?
Okay, fine. So you are saying that I would just select the window which is already selected and I
Mhm.
should just share. I got it. Okay. So let me just share it and please confirm that you are able to see my screen. Okay.
Okay, sir. Yeah, I can see it.
Are you able to see it?
Yes, sir. But I can see your whole window.
Okay. The PowerPoint as well, right now?
Yes, sir. Yes, sir. Right now. Yes, I can see it.
Okay, fine, perfect. So then I would stop sharing right now. Okay.
Okay, sir. No issue.
Okay. Thanks a lot. So now things are fine.
Yes.
So I shall join in a few minutes. I mean, I will be here only. I will just mute myself. Okay.
Okay. No, no issue. No issue. No issue.
Okay. Hi, Pankaj.
Good morning, Dr. Sandeep. Good morning, everyone. So we will be starting soon in 1-2 minutes.
Yeah, good morning. Good morning, Dr. Pankaj. So I hope my screen is visible, right?
Yes, you are audible and on the screen is visible.
So should I stop sharing in the beginning? Maybe I can share after you say I can start, right? That would be better, right?
This is fine. You can put the slides. I will be introducing you in a minute, and then you can start. Okay.
Okay. So I shall keep my video on in the initially and then I'll turn it off, right? So that would help the bandwidth.
Yeah, it does not matter. You can keep your video turn on or off. That is fine.
Okay, okay. Thank you.
Good morning, everyone. So let's start the proceedings. I would like to introduce our first speaker of the day, Dr. Sandeep Murarka. Dr. Sandeep will deliver a talk on HIT to lead in drug discovery. Dr. Sandeep did his BSc in chemistry honors in 2005 from Vidyasagar University, and then he earned his MSc in organic chemistry from IIT Bombay. Later, he also did a master's in science in organic chemistry at Rutgers, the State University of New Jersey. And then later he went for his PhD in organic chemistry. In 2013, he obtained his degree from University of Münster in Germany. After this, he worked as a Max Planck postdoctoral research fellow in the laboratory of Professor Herbert Waldmann at Max Planck Institute, Dortmund in Germany. He also served as a team leader in a reputed pharmaceutical company called Syngene International Limited for almost a year. Then in May 2017, he joined as an assistant professor at IIT Jodhpur. He has received Inspired Faculty Award from DST in 2016, and then also he earned Early Career Research Award from SERB in 2018. He is also a reviewer of several journals and publishers. His current research activities include study of novel activation modes and development of chemoselective and sustainable transformations towards synthesis of biologically relevant and interesting molecular architectures. So it's a great pleasure to have him here today for this session. So without further delay, now I would like to hand over to Dr. Sandeep. So Dr. Sandeep, please, you can start sharing your screen, and now stage is yours.
Okay, Dr. Pankaj, I hope my screen is visible.
Yes, your screen is visible and you are audible.
Okay, great. So Dr. Pankaj, thank you very much for your kind introduction. And I'm happy and privileged to take part in this Atal Academy sponsored FDP on multi-omics and data science, which is a great initiative to serve the scientific community and learn from each other. So today's topic of my presentation is Heat to Lead in Drug Discovery, which many of us are familiar with. So to begin with, let me just fetch my pointer so that it will be easier. Yeah, so to begin with, let's ask that what is a drug, right? So when we ask that what is a drug, that can be defined as a chemical substance that brings about physical, emotional, or any sort of behavioral change in a person, taking it. It basically, precisely, it could be any substance, it could be food, it could be water, it could be anything which alters or sustain some sort of physical, mental, or emotional state within us. When we bring this definition within the realm of drug discovery, which is precisely falls in the medicine which we are talking about today, then we say that a drug needs to have characteristics such as that should be of potential use in diagnosis or treatment of a disease. And a drug should obviously be selective in their actions, right? Because if it is not selective, if it is not supposed to attach to the target where we want it to be, then it will impart all sorts of non-specific interactions and their effect thereof, right? So when we go from this drug, but the question comes, how do we discover a drug? What is a drug discovery process? So the process by which new medicines are discovered is precisely known as drug discovery. A drug discovery process involves several interdisciplinary approaches. So in a nutshell, a drug discovery process, it's a heavily collaborative approach. It's not a one-man show, it's a teamwork where a multitude of people from across the disciplines comes on board, joins their hand together, and then involved in this project of drug discovery. People from biochemistry, molecular biology, organic chemistry, physical biochemistry, pharmacology, toxicology, cell biologists, and several other. The people who are also expert in the animal model studies, they all are on board in our venture to drug discovery. So it's often heard of that drug discovery is a very expensive and a long process. But the question is why it is expensive and why it's a long process. We'll go to that point, but before that, let's just try to understand that how did this all, how it all started, you know. So when we talk about drugs, then basically we talk about more or less two sources of drugs, right? It's a natural source or it's a synthetic source. So natural source means if there are various natural products which impart several sorts of biological and medicinal properties. For example, anti-cancer drug Taxol, which was discovered from a plant, and there are many other natural products which are isolated from, the active ingredients, rather to say, which are isolated from various natural products, impart diverse sorts of medicinal and biological properties, which eventually goes on to become marketed as a drug. Why synthetic chemistry is required? Synthetic chemistry is required because the amount which we obtain from natural sources are not sufficient to, to serve the billions of population which we have, right? And that's when a synthetic chemist, to be precise, organic chemist, medicinal chemist come into the foray to synthesize, to isolate the active ingredient from the natural product, which is actually imparting the activity of the drug, and then come out with an effective synthetic route to get to that compound, which is scalable, which is very important because we need to prepare that in a higher amount. Now, we all know the famous story of aspirin, which was discovered before the birth of Christ. You know, like before the birth of Christ, there was among the Greeks there was a saying that if one chews the bark of a willow tree, that can alleviate the pain during the childbirth, right? And then eventually a course of action took place. The active ingredient was isolated called salicine, which then was through several synthetic manipulations and alteration eventually lead to the development of aspirin, which was marketed by a famous German company called Bayer, right? So that's a prime example that how natural sources can guide us towards the active ingredient, which can eventually get marketed. Now, how does these drugs, be it from natural sources or from synthetic sources, act? So in this regard, I would like to invoke a statement by a famous German pharmacologist, Paul Ehrlich. He says that drugs will not act unless they are bound. So drug has to bind to a target in a specific way to impart the activity which we have desired for, right? And what are these targets? The targets are mostly these proteins, nucleic acids, and so on and so forth. I'll come back to all these points one by one in my subsequent slides. Now, let me put a pictorial diagram of a drug discovery process, right? In chemical terms, we can put a drug discovery program all in all into two parts or two ways. For example, a forward chemical genetics or a reverse chemical genetics. Now, let me first talk about what is a forward chemical genetics. In a forward chemical genetics, you start with an umbrella of compounds, right? A huge compound library, and you screen it in a signaling pathway which you are interested in. That could be Hedgehog signaling, that could be autophagy signaling, that could be any pathway which you are interested in. Then you screen the library of compound and you observe the phenotypic response. And then through this course of study, you will end up finding a compound which will probably be biologically active. Now you have found the compound which is biologically active, but then you do not know the target in the signaling pathway. So what you have to do, you have to do several studies, several mechanistic studies to eventually lead to the finding of the target. So basically in a forward chemical genetics program, you start with a compound without knowing the target. And eventually, after you find the biologically active compound, that guides you through a series of experiments to establish the target. Now, what happens in a reverse genetics program, which is the most modern and prolific approach? In a reverse genetic program, you start with the target protein. That means you know the disease, you know the target of which is causing the disease, and you have identified a good target. And now, since your target is known, now you decide a scale of compounds through either you know a standard and random screening approach, or you can opt for a guided approach, which you can refer to as a structure guided drug design as well. So through any of the approach, you get to a compound library. So in all, in both the ways, the prime thing is to establish a compound library which is structurally and electronically diverse. Because if it is structurally and electronically diverse, then only it has the chance to give you a compound which probably could be biologically active. Because if we want to cover a chemical space which encompasses as many as 10 to the power 60 compounds, then we have to have an educated guess about that compound, right? Because synthetic chemist, we can't reach to that level where we are making 10 to the power 60 compounds, because we have limitations with regards to time, manpower, and so on and so forth. So we need to start with an educated guess, and that educated guess will help us to build a compound library. Eventually, to build that compound library, what we need? We need robust synthetic methods. Because if we do not have robust and scalable synthetic methods, we can't build this compound library. And to develop those novel synthetic methods, we need to develop novel activation modes, which goes towards more about organic chemistry. So I would stop here. So basically, this is what is drug discovery program. Now to move ahead, there is a perception or there is a myth among all of us that drugs are always overpriced. But is this true? Is this absolutely true that drugs are overpriced, or they are overpriced, so to say, because there's a lot of cost is involved in the drug discovery process? So to understand that, let's see that what has happened over the years. So if you see that in the last 2 decades or so, what has happened is that the number of compounds, or rather to say NCE, new chemical entities, or the novel chemotypes, whatever you want to refer to, the number, if you see, the number has dropped down drastically. That means wherever you start with, the number over the years, the novel chemotypes coming out has been decreased. On the other hand, what has happened? The cost involved in the drug discovery process. So here I am referring to the total R&D investment, which starts with the initial target identification to the marketing, right? So the entire total R&D investment, what has happened? It has skyrocketed, right? So if the output is decreasing and the input is increasing, then definitely one can assume that this is absolutely expensive process. And we can easily see that there is a huge steep rise in the cost of drug development, whereas there is a decreasing number of registered products at FDA. So it is it is no big deal to say that drug discovery indeed is an expensive process, and it's a high risk process. Why it's a high risk? Because you can see that a drug discovery, to start with the target identification to bring it to the market, it takes as many as 8 to 10 years. So now when you are investing such a high amount for 10 years without knowing whether the drug will eventually come out in the market or not, then definitely it's a high risk process. Now let's try to understand that preclinical and clinical. I'll come back to these two terms in the subsequent slides. So for the time being, kindly consider these as two terms only. So preclinical and clinical studies, how much cost they impart in the year-wise or the decade-wise classification, if we have to see. So you can see that if you consider in 1970s, when actually the drug discovery started in the true sense, you could see that the preclinical costing was much more than the clinical costing. So basically, let me put it in a very, hazier word, that clinical means absolutely at the end stage of the drug discovery, where you are doing the trials on the humans, so that it becomes marketed eventually, right? And preclinical means all the development related work, right? So let me put it in this way, initially at least. Now, when you go to 1980s, it's similar kind of a C. Although overall cost has improved, you can see that the cost in overall millions have increased, but the ratio, as you can see, that the preclinical remains still high. That means the more amount is invested in the R&D rather than in the clinical testing or the efficacy and safety of the drug in the human trials. If you go from 90s to 2000, suddenly you can see the clinical costing. The overall costing has definitely increased, but the clinical costing has out it has outered than the preclinical costing. Now in the 2000 to 2010, the cost has improved drastically and clinical cost has really high. So to put things into perspective, let me give you a value that a prescription drug which you are taking that gets market approval at a cost of 2.6 billion. So that means in today's time, if you want to launch a drug in the market, starting from your target identification to the preclinical drug development to the clinical trials, and then eventually regulations and market, it costs 2.6 billion. So one can imagine how expensive this is. Now, if you go back in time, if you see in 2003, the costing was approximately 0.45 billion or so, you know. So there is a 145% of increase in the costing of a single drug launching in the market. Now the question is, why so much of price? Where does this money is going? Why drug discovery is such an expensive process? There are several folds of reason. One of the reasons is complexity of clinical trials, the greater focus on chronic and degenerative diseases, and tests on comparator drugs. So let me put this term in perspective, comparator drugs. So when clinical trials happen, generally they used to happen only against a placebo drug. Placebo means a drug which does not have any effect, the drug which does not impart any effect, right? So then that serves as a control. But nowadays what happens, because if there is already an existing drug for the same disease in the market and you want to launch a new drug for the same disease, then your drug has to be compared with the already existing drug. So it's a comparison between the newly launched drug or the drug which you want to launch with the drug which is already existing in the market. So that's called comparator drug, right? Now, because of these sorts of things, the complexity and the cost involved in the clinical trials has gone manyfold up. So again, to conclude that drug discovery is a high risk, expensive and long-term endeavor for sure. So we have understood that the drug discovery is an expensive process. Now let's go down, let's try to bifurcate this individual section and try to understand why this is why this is so much of costly process and where does the money is going, in which phase it is going in higher amount. So in drug discovery, generally you start initially with a very high pool of compound. It could go up to 100,000 to 150,000 compounds as well, right? And then immediately it cuts off to the initial stage to the several hundreds of compound. So the several 150,000 compound are involved in the high throughput screening. Then you get your initial hits. Then you get a lead compound, let's say around 5-6 compounds, and then that goes into the clinical trials. There are generally clinical trials are phase 1, phase 2, phase 3, sometimes in phase 4, and then FDA review, and then eventually it gets approved, right? So now you can see that the cost involved in the R&D, actually the R&D, not the clinical trial, is 30%. Whereas in the clinical trial, it goes about 50%. So the major chunk of the investment goes into the clinical trials. And I just explained in the previous slide in few words that why the clinical trials are exhausting a lot of money. Whereas in the manufacturing, as you can see, it's not much, 20%, right? So 40% of the compounds, and also there's a huge attrition rate, because you can imagine just to get that one drug, you are starting with 100 and 150,000 compounds just to get a one drug. So yet attrition is also happening gradually in each and every step. So now you can combine this cost of making these many compounds, the time and the money, the manpower and everything involved, and then the attrition which happens all the way to get to the one drug starting from 100 to 150,000 compounds, right? So that is why it's a very time consuming, long and expensive process. So it is not absolutely a myth that the drug discovery is an expensive process. I'll go through each and every step of drug discovery, at least from hit to lead, in my few subsequent slides. Now here, through this slide, I would like to give you a simple time frame that how much time generally happens in which step and what, like in a very summarized way, what we do in that step. So if we talk about the initial stages of the drug development, there we talk about target identification, means target discovery, which target I want to target to develop the drug. Then I have to validate the target, then to get to the compound, which I can take it to the preclinical and then subsequent clinical development. So before the preclinical development, I should have a lead compound which should have a drug-like properties and good physiological properties as well. Now, for this entire phase, till the lead compound optimization, it mostly takes 1 to 3 years. Finally, the lead compound which you have got, that you take it to the preclinical development for all the in vivo studies, as much as you can think of at advanced level of admit toxicological profiles and all and all. So there it takes generally 1.5 to 2 years. And then comes the clinical trial, which goes on and on for phase 1, phase 2, phase 3, submitting of several different sorts of approvals at different stages. I'll also talk about that. And then finally approval of the drug, which takes another 6 to 7 years, right? So that's where the 10 years of program which goes on. Similarly, if you put the cost-wise thing, then to optimize a lead, it takes more or less approximately around 200 million. In the preclinical development, another 100 million investment goes in. And then comes again the expensive part where the major chunk goes in the billions, 1.1 to 2.5 billion, right? That goes into the clinical trials. So with this thing in mind, and let me go back again. So here, as you can see that once you have the lead optimization, then it's a preclinical development and then clinical trials, right? So here what happens is that once you have a drug in your hand, then you have to file a IND, okay, and then it starts the clinical trial. So basically IND needs to be filed before the clinical trial starts. So I would not spend much time here in this slide, because the thing which we want to discuss today is the hit to lead, but not the clinical trials. But although towards the end of the talk, I'll just give you a brief overview what happens in which phase of clinical trial, right? Now, what is IND application? As I just talked about, so IND application is basically investigational new drug application. It's nothing but it's basically seeking permission to perform clinical trials. So when you have done your studies, you have done your preclinical studies and everything, and you have to submit all those data like table of content, your investigation plan, your pharmacological and toxicological profile of the compound, everything, basically every detailed study which you have performed, that needs to be submitted to the FDA and to seek your permission that you can start the clinical trials now, right? So that's the investigational new drug application. Now the trial can proceed if the FDA does not file a hold within 30 days of receiving the application, right? And the application must contain information on animal pharmacology, as I said, all investigation brochures, and etc. and etc. There is one more application which happens at the later stage, is called new drug application. That happens after the clinical trials are done. So when the sponsor of a new drug has enough evidence of the drug's safety and efficacy to meet FDA's guidelines and marketed approval guidelines, then the NDA can be submitted. So basically, once the NDA is approved, one can say that the drug can be marketed. Basically, at least this is the norm in the US. And the NDA application also has certain sets of guidelines that what are the documents need to be submitted, like like the non-clinical pharmacology, toxicology profiles, all the chemistry, manufacturing controls, all singles data regarding the synthesis, functioning, pharmacology, toxicology, medicals, every single data related to the compound needs to be submitted, right? So this is all about the introduction of the drug discovery process, which I wanted to give you a picture about, that how drug discovery happens, why it's a cost-effective process, and so on and so forth. We have also understood through these couple of slides till now that several different disciplines are required to begin a journey of drug discovery, right? Over 100 disciplines, as many as over 100 disciplines working together to make a fruitful drug and to make it commercialized. Medicinal chemist, among these chemists, so because it's a hit to lead, today's journey is about hit to lead, so I specifically chose the challenges which are faced by medicinal chemists. So medicinal chemists today, they face challenge because when the management decides a drug to be developed for a certain sort of disease, the cost and things are more or less fixed, right? And now since the cost is increasing in the clinical trials, so the cost in R&D investment definitely would go down, right? So there's an increasing cost and enormous amount of time taken to discover a new drug, and all the efforts in the synthetic stage are given by the medicinal chemist. And if the drug goes all the way in clinical trials and fails, so one can imagine the kind of disappointment it brings to the entire team, and as well as to the medicinal chemists who have made these compounds eventually, right? And as well as there are several big pharmaceutical companies like Novartis, Pfizer, GSK, several other companies, Novartis, they are all working towards probably the development of the similar sorts of drug or similar sorts of disease at least, right? And if the target also eventually comes out to be the same, then it's a fierce competition that who is going to launch the drug at a faster rate in the market. So this all load comes into the R&D team, that how fast they can bring a compound to the lead stage so that it goes to the preclinical development. So it's also a fierce competition. Now, having this stage set for the drug discovery, the cost involved, the stages involved, and so on and so forth, let's go back in time and try to understand that how it all started, right? So one can say that the history of drug discovery, it started in almost like a early 1900th century, where the first rational drug design started, right? What was the goal? The goal was to develop a safer treatment for syphilis, which is basically a bacterial infection. Initially, the compound which was used is called atoxyl, which is, by the way, a first organic arsenical drug used for the treatment for the bacterial infection, right? So Paul Ehrlich, which I say German pharmacologist, they wanted, or he and along with Hathard, wanted to maximize the toxicity to the pathogen and minimize toxicity to human, right? So they did a set of studies on these organoarsenic compounds, and they eventually find out an alternative compound which is known as salvarsan, which is again an organoarsenic compound, but which showed better efficacy as compared to atoxyl. But salvarsan also did not remain longer in time, because it was eventually replaced by penicillin in the 1940s, right? Then goes on to 1960, people started understanding the importance of SAR studies, structure activity relationship studies, structure property relationship studies, and QSAR. QSAR is quantitative structural activity relationships. So the first successful attempt to relate the chemical structure to the biological action. So what sort of structure or what sort of functionality or what sort of geometry of a compound is responsible for the biological action or is responsible for the effect which we are observing, that relationship? How can we quantify that, right? So all those things started coming into the picture, which led to a rational drug design. Now, once we move on, then we go to mid to late 20th century, people started understanding the disease states, the biological structures of the proteins, how they function, their 3D structures, and all those things. So once they started understanding these things, the detailed biological processes came into the picture, drug transfer, distribution, the ADME toxicological profile come into the picture, and medicinal chemists started using this knowledge to modify the chemical structure and to establish a structure activity relationship and structure property relationship correlation to enable the heat to lead advancement. For example, how little bit or the subtle change in chemical structure can lead to an altogether different drug with different biological or pharmaceutical property is a prime example at that point of time is procaine. Procaine is this compound which is having an ester kind of a linkage, and which is used as a local anesthetic. Once you change this linkage from ester to amide, keeping the other things intact, then it becomes procainamide, which is a completely different, it is an rhythmic drug, right? So that's how people started understanding that how subtle change in chemical structure can bring in a completely different therapeutic property of a chemotype. So what are the approaches one can use to the drug discovery? One can just believe on the luck, right? That fine. I can just do some random things and eventually I can probably end up getting a drug which is having a good therapeutic index. One can do this educated guess driven chemical modification or a random chemical modification. Screening, there are different sorts of screening. I'll come to that. And then rational drug design approach. So what are these terms? So when we talk about screening, we need to talk about the discovery of penicillin, which we all know, right? It happened in 1928 by famous Alexander Fleming. And the story is all of you know that how it all started, right? He went on a summer vacation in Scotland, then came back, see a messy lab bench. Then he saw that the petri dish has certain, the petri dish has some contamination with the airborne mold. He put those things under the microscope and he realized that there are uh, some sort of prevention of the normal growth of the staphylococci, right? And that's when he understood that this is uh, this has antibacterial property, and that's what led to the eventual discovery of antibiotic penicillin, which we all know. So this was absolutely serendipitous discovery by Fleming, which replaced many other this organoarsenic compounds as antibacterial drugs, which I already talked about. They were all replaced at this juncture by penicillin. When we talk about chemical modification, we talk about different approaches to change the structure or the geometry of the compound. There is a homolog approach. Homolog means increasing the number of carbon. So basically, if this is a compound where R connected with the amine functionality in between CH2 bridge. So if you increase the number of these methylene carbons in between, what can bring in the change? So this is called homologs of lead prepared. The N can be 1, 2, 3, 4, 5, etc., etc. Then molecular disconnection or simplification. So there are many natural products. There are many big compounds which impart biological activity, but we sometimes misunderstand that the biological activity which we are seeing is entirely due to the entire structure. But no, it's most of the times is due to a particular functionality or fragment. So if you can do stepwise, stepwise chopping of the functionality and to see that which part is imparting the activity, then you can actually come up with a simplified structure. So this is also a way to discover new drugs or new chemotypes. Similarly, instead of chopping it off, you can also do molecular addition. You can take two fragments and you can try to combine them to come up with a completely new chemotype to see what is the kind of biological activity it imparts. Isosteric replacement, in biological terms, we call it a bioisosteric replacement, is just a replacement of a functional group with another functional group, which is having similar sorts of property geometrically and electronically, so that it increases the it increases the solubility and other sorts of property of the eventual molecule. So that is the isosteric replacement. So these are all the things which are done to discover the new drug. Then chemical modification, which I already talked about, right? You can always think of chemically modifying a compound. For example, I will talk about this story in a few seconds. The cimetidine is a drug which was eventually chemically modified to a compound called Jantic and ranitidine, which you all may recall we used to take when we used to have an acidic problem, when our stomach used to create a lot of acidity. They are also known as hydrogen inhibitors, right? So screening. What is screening? So screening basically it means it testing a random and large number of different molecules and to just hope that we would come up with a compound which is having biological activity. So basically, as you can say, as I put it across, it sounds like a random screening, and it's exactly like that. So, for example, let me tell you that Prontosil is a drug which is having antibacterial properties, but this drug was discovered through a dye. And how it happened? So a German chemist and a pathologist, Gerhard Doma, he wanted to study the antibacterial properties of azo-dyes, right? And during his investigation, he find out that Prontosil, one of these azo dyes, actually impart antibacterial properties. So it was a part of a random screening process. So as I talked about screening, we got to know that it is mostly irrational and based on serendipitous discoveries, trial and error approach. It's time consuming because you do not know whether this kind of random screening would lead to something important or not, right? So then what can be done? So the next generation of drug discovery started through rational drug design. So rational drug design starts with a validated biological target. As I said in the beginning, that you know the target, basically it's like a reverse genetics program, chemical genetics program, right? You know the target and then you start. So for example, there was a problem that histamine triggers generation of lot of stomach acid. So people wanted to develop an antagonist of histamine to suppress the generation of the stomach acid, right? So here histamine is the target which already is known. So through a series of studies, they discovered N-guanyl histamine, which is also known as a hydrogen blocker. So this served as a lead compound that, okay, fine, now I have a lead compound in my hand, which is N-guanyl histamine, which can actually suppress the generation of stomach acid by histamine. And then you can see that how this compound, which is N-guanyl histamine, went through a series of rational designing to eventually lead to the generation of a drug, which is called ranitidine and Jantek, which you all know. So first you start with N-guanyl histamine, then it was replaced, the guanidine part, the basic part, where it was replaced with a excuse me, it was replaced with a thiourea kind of functionality. But what was found, that thiourea congener, although it imparted more potency, but it has got its toxicity in the clinical trials. So then this thiourea fragment, which was responsible for the toxicity, was replaced with another functionality, aminonitrile functionality. And then the drug became known as cimetidine. Eventually, cimetidine went up to the development of another drug which imparted more efficacy and as well as safety, is called ranitidine. So this is called rational drug design, which started through a known target, right? So when the drug discovery thing started in 1970s, it started mostly with the two-dimensional approach of the compounds. Now, as we can imagine, it is not a rational thing because you can't, these are not fixed things, because protein dynamics, protein function, everything is dynamics, right? So you cannot go ahead with a lock and key kind of approach. So What happened? Later stage, what happened? People started understanding about the 3D parameters coming to the picture, the compound's 3D structure, all this bioinformatic, chem informatic, virtual screening came into the picture, computer-aided molecular design came into the picture. So now all this advancement happened in the second generation. Then what happened? One more important thing, in the second generation, people used to always understand like a lock and key model, right? For the drug and receptor binding. But later, it was also realized that the drug receptor interaction is not a static, so you cannot consider a lock and key approach, right? Rather, people started talking about an induced fit model where when the drug binds to a target, the target is able to change its geometry, right? So it's not a simple lock and key. And mechanism-based drug design, as I said, which is a modern approach where you know the target, you know how the target is actually imparting the signaling, how the target is causing the disease, all these things are known. So mechanism is known. So which is more rational and disease-driven approach. So whatever we have talked about in the second part of our drug discovery today, the historical evolution of the drug design in the ancient times, like the natural products and then biological activities, then the chemical era, which involves random screening, then rationalization of the design process, and then in the biochemical era, where you start with biochemical high throughput screening on the target which you already have established, right? Now let's go through each of these drug discovery steps and briefly learn about those steps, right? So the first step is target identification. So what you do in target identification stage, basically you identify a target. If you do not know your target, then how do you go ahead? So first thing is you have to know the disease, right? And then you have to basically capture the clinical spectrum of the disease and the exact role of a potential therapeutic target. So once you have identified a target which is causing, then you go ahead with the target validation. You have to validate the target that indeed this is the target which is causing the disease, right? Because we know that drugs will not act until they are bound. We have learned that, right? So now move on. So there are several ways of validating a targets, right? There are many approaches one can take. Once your target is validated, then you have to screen a library of compounds. And generally, these are done through high throughput screening. Although there are different screening approaches, but mostly they are done through high throughput screening. Now, to do a screening, you have to develop an assay, right? So that's called high throughput screening assay. That assay can be a biochemical assay, that assay can be a cellular assay. But important thing, that assay should be good for the target. Your assay should be such that it is able to screen a large number of compounds in a shorter period of time in a cost efficient way. So once you develop the high throughput screening assay, through that assay, when you screen 150,000 of compound, then you can get to the initial hits, which are a couple of hundreds or thousands. Then you take those hundreds and thousands of compound and do the heat optimization through SAR studies. Then you get again a small 100 collection of compounds. Those are called leads. Those 100 lead compounds, again you do a smaller SAR and SPR, which is a structure property relationship studies, and then those compounds become good enough for the development and eventually to the preclinical and clinical trial, right? So let's first talk about target identification. So a biological entity. What is a target? A target is nothing but a biological entity, which usually a protein or gene that interacts with and whose activity is modulated by a particular compound. That's how the Oxford Dictionary of Biochemistry and Molecular Biology defines a drug target, right? So what are the properties of drugs a target should have? A target is any system that can potentially be modulated by a small molecule, right? And a biological target of a drug can be proteins or nucleic acids, right? It mostly these targets are receptors, ion channels, GPCRs, and EGFRs and kinases and so on and so forth. Mostly they are proteins, nucleic acids, and so on and so forth. The drug can be small molecule or the biologic, right? The target should be druggable. That is a very important thing, that your compound should be able, or the small molecule or the biologic should be able to bind to the target. So that means the target should have either a fixed pocket or a allosteric pockets, right? Druggability is the ability of a compound to bind with high affinity. So when a compound is able to bind with a target with high affinity, then we can call that compound a drug, right? So this is about target. Now, what are the important attributes of a good target? The important attributes of a good target are: the target should have an established role in the physiology of the disease. So that means if I have identified a disease and thereafter the target must have a critical role in the disease. And the target's 3D structure should be available to assess the druggability. As I told you that today we have to talk about the 3D structure, right? So the targets, the protein which you are talking about, the protein's 3D structure should be available, because otherwise it would be difficult to do a computer-aided drug design, the virtual screening, or the structure-guided drug design, right? The target should be easily assayable, means you should be able to develop an assay for the target in an easy way. And the target should possess a promising toxicity profile, means all the relevant toxicity profile, the toxicogenomics of the target and everything should be well known. And of course, if when a pharmaceutical company goes on to develop a drug, if they discover a new drug target which was not known before, then definitely the IP value goes up, right? But then of course it is also a higher risk driven. So it depends on the company to company that whether they want to go ahead with a new known target, or they want to go ahead with absolutely new target, right? Then goes on target identification. So what is target identification? So target identification is basically identifying a biological target that is druggable, right? So when you are done with the target identification and you have established the role of the target in that disease and so on and so forth, then you move ahead. You move ahead to the step where you want to check that the drug which you have want to develop against the target, is it efficacious, is it safe, and whether the target is also, there are many targets which you if you develop a drug against that target, sometimes it shows more toxicity profile also. So you have to see that the target is such that it should allow you to develop an assay and a compound of thereof which is safe, right? So in that way, as I said, there are few popular and well-known targets which are found to be well established, and their toxicology profile and as well as the other profiles are well known, such as GPCRs, kinases, EGFRs, and so on and so forth. So there are two ways one can go ahead with the target identification strategy. One is target deconvolution, the other one is target discovery. I have more or less discussed this in the very beginning. Target deconvolution is nothing but a forward chemical genetics. In this scenario, one begins with the efficacious drug, and then basically, how do you get the efficacious drug? Through a random screening of the compound library, right? So you start with the efficacious drug, and then you try to find the target through different means. Whereas in the target discovery, what happens? Your target is established first, your target is identified, and then compound libraries are screened to find a drug that binds to the target. So it's the reverse chemical genetics. So in the forward chemical genetics is basically a target deconvolution, and in the reverse chemical genetics, it's the target discovery. So I have pretty much said the same thing which I have just discovered. So I'll skip that slide, and I'll go ahead with the next stage, which is the target validation, right? So in the target validation, there are several ways of doing the target validation. But what we do in the target validation, we basically establish the compound against the target in a physiological, pathological, and pharmacological way, right? We demonstrate that the functional role of the identified target is indeed exist, and is causing the disease phenotype. So it's also in a way of risk assessment, right? That my target is fine, it doesn't cause much risk, there are not much side effects, and it has got good toxicity profile to go ahead further for the drug development and so on and so forth. And initially, once we evaluate all this risk, the better it is for the project to advance at the later stage, right? And the target validation can be done on a molecular, cellular, and sometimes on a whole animal level as well, which is not advisable, right? There are several methods can be used for the target validation, right? And it can be affinity chromatography, pull down, knockout experiments on the genetic level, and affinity chromatography. And so these are all biological foray where biologists do separate sets of studies to do the validation of the target. This is a small table which says that affinity chromatography generally is used in target deconvolution approach. So when you are going with the target deconvolution approach, generally you go ahead with the affinity chromatography and so on and so forth. I'm not getting into that at this point of time. To summarize the target validation part, one can say what Dr. Killian VM Hoover said, that the only real validation is if a drug turns out to be safe and efficacious in a patient. A major challenge is that many targets fail late in the clinic and several years after initiation of the project. Thus, we have to improve our ability to identify promising targets at the early stage, and open science and sharing of data map help in this regard. So this is all about target validation. So once we move ahead from the target validation, we go to the high throughput screening, heat, and lead. So there are several screening approach: high throughput, focus screening, fragment screening, virtual screening, but I'm going to focus on high throughput screening only. I'll skip this slide because this is basically talk about what we have just understood, that you have to get the target identification, and then through several series you do in vitro studies, assay development for the high throughput screening, then you get several compounds, and then if the compound goes on, shows drug-like property, you move ahead to the lead optimization and clinical trial. If it fails, then again you have to come back to the compound screening library, right? So what is high throughput screening? So high throughput screening is a screening where you have established your target and you screen a large number of compound, as much as 150,000 compound against the target to get some compound which has got initial some sort of biological or drug-like property, right? Once you get the compounds with desired effect on target, they are called heat. So high throughput screening is a process enabling finding of heat. If you find a heat, then heat directly used as chemical probe to study target, or heat provides structure for the chemist to begin developing a drug and go to the stage of lead, right? So it's like a funneling approach. You put a lot of compound in, and then eventually from several 100,000, you get either a couple of thousand or a couple of hundred compounds through high throughput screening. So to keep things in perspective, if you see the diagram of the high throughput screening, you can see that several 100,000 compounds are taken. Compounds are collected, you develop a high throughput screening assay. It could be, as I said, biochemical assay or cellular assay. You screen those compounds against those assay. You find the activity, that's the initial hits. Then you have to re-establish the effectivity, right? That whether they are actual hits or some kind of some kind of random results. So then you have to establish some sort of parallel assay to again screen those hundreds of compounds which have found the hits to understand whether they are false hits or not. Once you screen those compounds again against the assay which you have developed, which are called parallel assay, then you have confirmed their activity, so they are real hits now, right? So you take those hits, you do the SAR studies and all other structural conformation and variation to go ahead to find out the qualified hit list compound to go ahead to the lead stage. So up to this improved hit list, it's you get through the high throughput screening. So I have talked about heat and lead a lot as of now. So let's just put things into perspective and understand this term. So heat is basically a very preliminary positive result. They are not expected to be highly reactive or highly, active. They can have low specificity and low efficacy as well. It can be in the millimolar range as well, if not micromolar. And then they are often obtained through high throughput screening, right? And whereas on the lead is little bit optimized. It's a suboptimal drug, but it's still not a compound which can be taken to the preclinical development. It needs to be modified to better fit the target for efficacy and as well as off-target activity and specificity. So this is heat and lead. So this is what happens in the high throughput screening, which I have already talked to you. So I'll skip that slide. We need to understand that when you go from heat to lead, already it's only 40 to 50% goes on, right? So as I have talked to in the beginning, that the chemical space is huge, constitutes of 10 to the power 60 compounds, whereas a corporate compound library is only 10 to the power 6. So one has to build a compound library based on educated guess and based on the inspiration taken from nature, right? So that we are more closer in terms of the in terms of the drug like chemical space, because more closer we are, that increases the chances of hitting a compound which can eventually go as a lead. So once you have the heat, once you have validated the heat, then against the functional assay, which I have said it's a parallel assay, then you can go ahead to the lead stage, right? So I'll skip this slide again. So who are the people who are generally involved in the high throughput screening campaign? Definitely biologist, molecular biologist, automation specialist, because high throughput screening is an automated process. You need a chemistry help, IT help, because if you are doing a virtual screening, if you are doing a high throughput screening in an automated process, then definitely you need IT help, right? And chemical library help to manage the data, right? Which is the chem informatics. To manage this much of chemical data and chemical library, you definitely need to have a library kind of forum. Then you move ahead to the next stage, which is the lead identification. So you have got the validated hits, and now you have to identify the leads out of those validated hits, right? So at this stage, the validated hits would be tested to determine factors such as selectivity, whether it is selective towards that receptor or it is having many off-target activity, right? You have to see the physiochemical properties, the initial admit toxicological profile of the compound, whether it has drug-like properties like the cell permeability, solubility, and there is a Lipinski rule, I'll talk about that, and then other metabolic properties, right? So you see all these things when you are developing a lead. In the lead stage, you need a hardcore medicinal chemist, because a lot of structure property relationship studies involved in this stage, and ADME personnel, so the people who are expertise in doing admit studies in the mice model. Nowadays, let me tell you also that admit studies can be done virtually also, right? So to save the cost and a lot of things, people are doing the virtual admit and toxicological profiling before they actually go to the lab. So everything is becoming virtual, as not only what we are doing now, virtual seminars, but in the drug discovery also things are happening more and more virtual now. People want to do more things in the computer before they go on actually to the lab, to save the time, resources, and definitely the cost involved in the drug discovery process. Now, coming back to the ADME again. So what is ADME? ADME is basically absorption, distribution, metabolism, and excretion. How well, when you take a drug, how well it is absorbed, how well it is distributed in your system, metabolized thereafter, and excreted out of your system. All these four parameters are absolutely essential. Now, methods to determine potential biological activity in an oral drug is basically ADME. This is also known as Pfizer's rule of 5. What are the rule of 5? Number of hydrogen bond donors. So when you are making a compound, the compound should have certain attributes to make it a good drug. But it doesn't mean that if you have these attributes, it will eventually make a good drug only. Despite fulfilling these criteria, the drug may not be good. But what are these rule of thumb or rule of 5? It says number of hydrogen bond donors should be no more than 5. Number of hydrogen bond acceptors should be 10 or less. Molecular weights should be less than 500 Dalton. And clog P, which is basically the partition coefficient between octanol and water, is a measure of lipophilicity. So your compound should be lipophilic enough, right, to go to the cell membranes and all. It should be less than 5. So this is called ADME rule. This is also a part of pharmacokinetics. It all comes in the toxicology, right? So now, so ADME and toxicology happens together. So you see the ADME properties and also you develop a toxicological profile of your compound, that whether how much toxic it is. These are all mostly happen virtually, and then it goes on to the mice model and rabbit model, right? Characteristics of a lead molecule. Generally, a lead molecule will have a greater affinity potency than a heat, and it will be tested against a lot of target to see whether it has an off-target activity or not. It should have acceptable physiochemical properties, which we have just talked about, right? And it should not have unwanted properties such as toxicological or physical. I need to little bit rush through here because I have slides couple of where I want to give you example of a real drug development program. So I will go through this introductory shares a little quick here. So then next is lead optimization. I have already talked about here. So lead identification and optimization, 2 stages, right? From late stage heat, I get a lead compound. Then I take that lead compound, screen their physiochemical properties and ADME properties. Then I get the optimized lead compound, which goes to the preclinical development, right? So at the preclinical development stage, all the in vivo studies, the bioavailability, the how it should be injected, how it should be taken up, the oral or the intrapenetorial or intravenous, all these sorts of solubility, all these things are taken care of in the preclinical development stage, right? But what happens sometimes when you start with a lot of heat compound, but still you are not getting the lead? So that's a high throughput screening fails. So that's why nowadays people always try to first do virtual screening, molecular modeling approach to see as much as closer hits so that you can take it actually in the lab. So before you take it to the lab, you do a lot of computational work. Similarly, to do the actual in vivo studies of ADME and toxicological profile in mice model, nowadays people do it on the computer. You can do a virtual early ADME and toxicological profile to find out the potential metabolic hits and metabolites, so that with that educated knowledge, you can go ahead to the in vivo studies. So these are all alternative studies. So as we have understand, it's a subtle balance. It's which is keep on changing. Sometimes you have to deal with activity optimization, which is like the structure activity optimization. Sometimes you have to do property optimization. So generally in the heat stage, you do SAR studies. In the lead stage, you do structure property optimization studies, right? And this balancing goes on between the properties and activity. So we have understand that drug discovery is actually a juggling act, right? You have to juggle between the different activities and different properties all the time. Sometimes solubility, stability, selectivity, activity, how safe it is, pharmacokinetics, and so on and so forth, right? Now, clinical trials, I would skip that part because otherwise I would not have enough time to go to the example which I want to give, but I'll just quickly give you just what happens in clinical trials. So once a candidate has been selected in the preclinical development stage, then the various safety hurdles addressed, it can be entered into the clinical trial. So once it enters in the clinical trial in phase 1, then the things start. In phase 1, what happens? Generally focus on the bioavailability and pharmacokinetic properties, not the efficacy or toxicity, right? That's what happened in phase 1. The drug is administered to a small number of healthy volunteers in the trial participants. In the phase 2, what happens? Mostly focusing on the efficacy of the drug in a large number of patients, right? It could be several hundreds as well, who are already having that problem, right? So efficacy in the second phase in the clinical trial. Third phase, if you get the enough good result in the phase 1 and phase 2, then your drugs move ahead to the phase 3 clinical trial. In phase 3 clinical trial, even it happens on a larger versions. And then you have to answer specific questions with respect to efficacy. The trials would routinely involve several thousand patients from different regions, different places, and specifically to the people who are currently undergoing the treatment. And here also your comparator drugs things also come in, right? which I have already discussed, that if there are any drug in the market which is already showing, which is already in the market, that needs to be compared with the drug which you want to launch. Phase 4 may or may not happen. These trials are often referred to as post-marketing studies. You know, they are basically to take the feedback, what happens. This give a greater idea of long-term risk assessment, benefits, and so on and so forth. Whatever we have discussed, let's summarize through a pictorial diagram: drug discovery and development. You identify a disease at the outset, right? Then you try to find out a valid target which is causing the disease. So you isolate the protein, you identify the target. Then you find a structure, if you can possible, find the structure of the target protein. Preclinical testing using a drug which you have found out. You have to scale it up to do the preclinical testing, because it happens in a mice model, in vivo studies. Once it's done, you go ahead to the further stages. You have to find out how the formulation can be done, how the bioavailability of the drug is, whether it's orally available, intraperitoneal, intravenous, all these things. Once it is done, then you file IND to ask the FDA that you want to go to clinical trial. Then the FDA gives you approval for the clinical trials. Clinical trials happen in 2 to 10 years. And then once the clinical trials are passed through, then you file NDA. That is, you want to market the drug. And finally, the FDA gives the approval, 2 to 3 years. This is the overall process. Now, I want to show you that how things are becoming, how things are changing now. So when I talked about at this phase, we did not talk about detail in drug design and development. There are two parts, design and development. Nowadays, the design happens through molecular modeling and virtual screening mostly. Before it was through random screening, but now it's happened molecular modeling and virtual screening. And that's where the chem informatics come into the picture, which is a huge database of the chemical compounds with their properties, right? How the technology is impacting the drug discovery process and which stages the technology can pitch in, which I have talked about. When you isolate a protein, that's where the genomics, proteomics, and biopharm come in to understand every single information related to the protein, right? So potentially producing many targets and personalized targets. You have to see whether what you are producing, is it a valid target, is it a personalized target, whether there are alternative targets are also possible for the disease and all. The better information. So all these things are today done by genomics, proteomics too. High throughput screening, which is again an automated process. You need a lot of IT help. Initially, you have to use a man-made manual things, but now hundreds and thousands of compounds can be screened in no time to get to the heat compound, right? This is all automated. Before even automated screening, after automated screening, once you get the initial heats, before you get into the lab, make the compound, now people do virtual screening to cut the cost, cut the time, and improve the efficiency, and reduce the attrition rate at the later stage. So this is virtual screening, combinatorial chemistry to make the compound library and all. Molecular modeling, docking studies have come in, which comes in mostly in reverse genetic approach. And then in vitro, in silico ADMET models, as I told you that ADMET models and toxicological profile now all can be done in silico to again save the cost and all. So that's how the drug discovery has become today. So I tried to give you a broad spectrum of the drug discovery and development right from the history till today, right? And how the technology is coming into the picture, how computer-aided things are actually revolutionizing the drug discovery process, and eventually, hopefully, it will also cut down the time and cost involved with the drug discovery process. So this is the last part of my today's talk, where I want to give you a case study that how I was involved in a drug discovery approach. And I want you to think of this, to think of the introduction which I have talked about while you are hearing me out in this case study, right? I will try to correlate as much as I can. So as I said, drug discovery is a highly collaborative approach. So this is a collaboration at Max Planck Institute of Molecular Physiology between 3 departments. One is Department of Chemical Biology, which is headed by Professor Herbert Waldman. One is Systematic Cell Biology, headed by Professor Bastian, who is involved in all the cell-based assay development. Then the Structural Biology group, who do all the structure-guided drug designing, all the molecular modeling, the co-crystal structures, the crystallography involved, all these things are done by the structural biology group. There is one more thing which is not done here, the animal studies, which was done by a small company there, right? So as I said in the beginning, the drug discovery is a field where people from across the disciplines, across the expertise come and join their hands. So it's a highly collaborative teamwork. It's not a one-man show, right? So this is the collaboration. So what is the target they wanted to, what is the disease, let's say, what they wanted to address? They wanted to address cancer. Now, RAS proteins are one of the old proteins or one of the first proteins isolated to show that they have the oncogenic properties, right? So RAS proteins, basically they functions as a binary molecular switch that cycles between an active GTP bound and GTP bound states, right? The GTP bound is the RAS gets GTP bound when it gets the extracellular stimuli that can be anything. Then from the GTP bound state it goes to GTP bound states, and in the mutated RAS form it remains permanently on, right? Now once it is remained permanently on in the mutated RAS form, then it leads to the cancer because of uncontrolled signaling, right? So this is RAS protein. So generally RAS proteins are, the RAS family of proteins are several, like HRAS, KRAS and NRAS. But out of these three RAS, the KRAS has been responsible for 30 to 40% of human cancers, especially colorectal cancers, right? So this collaborative team became interested in addressing this issue and targeting KRAS proteins. But then the problem is there are direct inhibitors of the KRAS proteins were not known because KRAS surface has almost flat. It does not have, it is not druggable. At least that was the belief. Since very recent discovery of Kevin Sokot, where he discovered that the allosteric inhibition of the G12C can be done. Nevertheless, at that point of time, this collaborative team, which I have just talked about, they identify another protein, which is called PD delta, phosphodiesterase, the delta unit of phosphodiesterase protein. What does this protein do? Now, it was understood that the RAS undergoes several post-translational modifications like many other protein do. One of the post-translational modification is farnesylation, that the RAS get attached to a farnesyl group. Why this is important? This is important because the RAS protein imparts its activity in the cellular growth and all other thing when it is bound to the plasma membrane, right? So after the farnesylation in the cytoplasm, the intracellular trafficking of the RAS protein is actually modulated by another protein called PD delta. So PD delta act as a cytoplasmic chaperone of these farnesylated RAS proteins and are responsible for the intracellular trafficking. So what was thought that can we target the PD delta? If we target the PD delta, then PD delta will not be able to transport the RAS protein from cytoplasm to the plasma membrane, and in turn, it would lead to the RAS mislocalization. And that's how a target was established to suppress this oncogenic RAS signaling. PD delta was established as an important target to suppress the oncogenic RAS signaling. So this was the target identification. I am not talking about the target validation because I can tell you that target validation was done easily by a drug called, let me recall, it was Taxol. So Taxol was labeled, and Taxol was already known to bind PD delta, right? So through a study, it was PD delta was validated as a target. So target identification, validation done. The next stage, if you recall, was the high throughput screening. And I told high throughput screening can be done through biochemical or cellular assay. So the high throughput screening assay was developed using alpha screen technology, which is a proximity assay, right? You can read about that alpha amplified luminescent proximal homogeneous assay screen. So what happens here? You have a donor bead, you have an acceptor bead, and when you excite the donor bead, there's a singlet oxygen transfer happens, and through a series of luminescent signaling, what happened? You see the luminescence, and luminescence is recorded as the output thing. And what happened in the proximity assay? If your donor bead is attached to the KRAS peptide in this particular case, and the acceptor bead is attached to the histidine tag PD delta. So the PD delta is your target, KRAS peptide is your peptide-based drug in this screening process, right? When they bind together, at least to 20 nanometer or so, then only, sorry, 200 nanometer or so, then only you will be able to see this signaling. If they are not binding, if their binding is suppressed by means of small molecule inhibitors, which you have subjected in the compound library, then you would not see the luminescence. Basically, this is the proximity ligation assay, right? So once you do that, then you will be able to get a lot of hits. So that's how we did also. We also got low nanomolar potency compound, which is pyrrolopyridazinone and pyrazolopyridazinone based scaffolds. These targets were validated. These initial hits were validated by fluorescence polarization assay. Remember, I talked about parallel assay or the secondary assays, right? So these are called those kind of assays. Now, initial these hits, pyrrolopyridazinone and pyrazolopyridazinone, were taken up one by one to go ahead to the lead compound. First, the pyrrolopyridazinone was taken, and then this compound was synthesized in several. I am not talking about that. Let me just quickly go through that. This compound was synthesized, and several compounds, as many as 100 compounds based on different structures and geometries, were synthesized. Initial screening find out that the amide-based pyrrolopyridazinone imparted good nanomolar activity, but they were not so good enough. So we wanted to replace the amide with the corresponding bioisoster, which are called oxadiazole, this is oxadiazoles, right? Once we did the oxadiazole, the affinity did not improve, but at least we were able to get the first co-crystal structure of the compound, one of the inhibitor and the PD delta protein. Now, because of that, what happened? It allowed us to do structure-guided drug design. So in the co-crystal structure, what we find out that the compound goes deep into the pocket and forms a hydrogen bonding with the arginine, and towards the end of the protein pocket, it forms another hydrogen bonding with the tyrosine. So these two are the hydrogen bondings, other than the other hydrophobic interactions were involved in the binding of the drug target to the protein. Now, based on this understanding, when we did the docking, we realized the northwest corner of the protein pocket is quite empty, where actually it can reach out to have enough hydrophobic binding. So with that limited knowledge, what we virtually kind of designed a molecule that can be have a N-alkylated, N-benzylated benzimidazole, so that we can fit the compound in the protein pocket in a much better way, and that should improve the efficacy of the drug. We went back to the lab, we synthesized the model compound, which is N-benzylated benzimidazole and N-thiophenylated benzimidazole, and we test their efficacy, and indeed we found out that these two newly synthesized compounds are having high activity as compared to their corresponding oxadiazole motif, and imparted activity of very low nanomolar activity. So this compound would serve as a advanced heat compound. So this is one example how to get the advanced heat compound enabled by structure guided drug design. Now let me take you to the second class of compound, which actually we took it to the lead development, is the pyrazolopyridazinone. Here also initial heat gave us a very good activity, and the binding pattern was same, arginine and tyrosine hydrogen bonding was involved. This is a zoom up picture to show you that how tyrosine and arginine is replaced. If you see here, there is one more protein, which is glutamine, is also nearby proximity, which also has a hydrogen bonding. Now, when we prepared the scaffold, we prepared a compound library, and then we went ahead to the SAR studies, structure activity relationship studies. And I'll just sum up by saying that we found out a compound which is highlighted here in the sky color box with a very low nanomolar affinity, which we taken up, which we decided to take the further lead development because of several reasons in terms of solubility and so. This is the initial early ADMET profile, which is pretty promising, having a good binding to the plasma, having a good pampa reflux, also, and other Kappa profile. I'm not going to talk about that. So in detail, basically Pampa is the parallel artificial membrane permeability assay. It's actually when the compound goes into our body, it has to cross some mini membrane, barriers. So it has to have a good permeability. So all these values are good enough, well taken, except one value, which is the intrinsic clearance rate in the mouse liver microsomes. The rate should be very small. But the good thing is, whereas although one of our compound 93 showed high clearance rate, the clearance rate drastically decreased when we went ahead to the other two compounds. I will show you immediately which one is the structure. So in medchem studies, there is something called metabolic hotspot. The benzylic positions are very prone to oxidation, right? So in the 93 compound, you can see that the benzylic CH bond are replaced in 99 with the fluorine. As soon as we replace this, we found out that the clearance rate decreased drastically. That means the drug is now having a higher lifetime in the mice liver microsome, right? So this is a very good thing which happened. So at this stage, having these results in hand, we decided to take this 99 compound to have the pharmacokinetic and toxicokinetic studies. So now we are taking this compound even after structure property relationship to the development stage. So then this compound was taken and then it was tested in vivo studies. So oral viability was not good because the plasma membrane concentration of the compound was very less. Eventually, it was taken to intraperitoneal, but that was also not very good, and higher loading was not tolerated. The toxicity started kicking in. And finally, what was found that the intravenous injection was optimal. In case of intravenous injection, it showed better results, right? Which is in the next slide. I'm sorry, I'm a little bit rushing through to give you a clear picture because anyways, this is not part of today's thing because we are talking about the lead till the lead development stage, right? So PK profiling of pyrazolopyridazole, one of the compound 93, which showed initially the high clearance rate was not so good, but then 99 was shown to be quite good. So once an IV application was better than IP and oral administration, the composition was found out. So that's where the project was stopped, right? So to sum up everything, I have tried to give you a picture of the drug discovery from the initial stage till the development, preclinical development, and I have tried to give an example, real example as well, and have tried to correlate each of these stages with the drug discovery stages which I have talked in before. What we have talked about, let's have some couple of observations out of that. So over the past 20 years, there is a number of changes in the pharmaceutical industry which I have tried to portray in my slides. The black box discovery process has now got translated to a mechanistic approach, which is a target-driven approach. There are many advantages of this approach, but then there are some disadvantages also. Now it has come down to the level where you talk about one disease, one gene, which actually has limitations because you are not thinking that one target can be actually responsible for many kind of diseases as well, and one compound actually could be good for multiple sort of diseases as well, right? For example, disorders such as schizophrenia, where effective drugs seem to target multiple receptors, right? Other thing is that as our knowledge about disease is increasing, so is our knowledge about the potential off-target activity of the compounds. Now, because of this, the regulatory burden is increasing, the chemical space is decreasing, which may account for the reduction in NC is coming in. Because what does this mean? More and more our off-target activity knowledge is going in, more and more we are becoming critical about the compounds which we want to take it forward, right? And because of that, there is a high level of activation is happening in the NC is coming in. NC means novel, new chemical entity, basically new compound. As the procedure of developing drug becomes more industrialized, the place for the maverick drug hunter becomes threatened. Maverick means non-judicious approach with an open mind, and you just go ahead. Like, so now people are having more judicious, educated approach, which is good in a way, but the serendipitous things which used to happen is also coming down, right? So off the wall ideas. So basically, sometimes we come up with a very radicalized ideas or so on and so forth. The scope of those radical ideas are also coming down. So everything has its pros and cons, but I can tell you in a nutshell that in today's time, the drug discovery approach is happening in such a way that the mechanistic and target driven approach, the reverse kinetic chemical genetics, is much and more popular, and it is actually probably the future of the drug discovery. And especially with the computer aided tools, the virtual screening and everything coming into the picture, hopefully the drug discovery process will become more amenable to time and as well as to the cost. So with this, I conclude my talk. So thank you very much for hearing me out. I'm sorry I took a little bit longer and less time for discussion, but I shall be happy to have any questions if you have any. Thank you very much.
Thank you very much, Dr. Sandeep. It was very wonderful talk.
Thank
So
you.
let me see if there are any questions from audience or from participants. So I request participants to write their questions in the chat box, or if they want to interact with Dr. Sandeep, you can raise your hand, then we can promote you to panelist and you can ask.
So do we have any question?
I'm checking in the chat box. So far, there are no questions, Dr. Sandeep.
Okay, so what is the I think I have a question now. So what is the name of the element component of the drug that makes the side effects of an administered drug has the okay. So it is not exactly clear. So basically, when we administer a drug in the body, so it goes to the admit and toxicological profile, right? So the drug goes through the metabolism states and it generates several metabolites, right? So most of the times what happens, these small metabolites end up giving the toxicity, right? But nowadays the good thing is you can actually do, as I have showed, you can do early admit toxicological profiling in silico. So once you do this in silico admit toxicological profiling, you have an educated guess about the compound. So you can modify the compound beforehand and you can actually generate those metabolites in a virtual mode. So you can understand the toxicity imparted by this compound even better way, right? So to answer this actually these are the small metabolites generated in the metabolism stage of ADME of the compound. They are actually causing the toxicity to the compound. Yes. And sometimes also there are some functional groups which are not advisable to have it in the compound. Those functionality as well cause the toxicity. For example, sulfur containing compounds are not very much advisable because of their bad smell as well. So people try to clear these things in the early stage as well so that they become better.
Thank you, Dr. Sandeep. And I think there is one participant who wants to interact. Rajesh Anand.
Yes, yes.
You can unmute and interact with Dr. Sandeep. Yes.
Sir, good morning. Thank you very much for the beautiful presentation.
Thank you.
I have a couple of questions. Thanks for the first question. Actually, my interest is I like to use the aspirin because it's a very well-known, one of the well-known drugs. But the problem is in colorectal cancer, the people, they're not encouraging, especially the clinicians, because it's making side effects of internal bleeding. So I wanted to know but there is one more information I like to share, like in Europe and in US, there are so many clinical trials are already going on. There are multi countries, they're publishing high journals, everything, but still FDA is not approving because just one reason, that internal bleeding. So could you suggest to me what is the name of that particular element that makes it internal bleeding? I'm not from chemist, so that's why.
Well, actually, to be honest with you, I'm also not aware of that. That is, what is the particular element which is causing the internal blood bleeding, you know?
Okay,
So
okay.
it would be interesting for me to also check it out. But I would assume, I mean, I just would like to guess it, that sometimes, you know, what happens, it becomes very difficult to pinpoint at certain compound, you know?
Mm-hmm.
because as I have said, that what happens once the drug goes into the body is very difficult to understand. It could be that this compound which you are talking about, it could be some sort of degrading metabolite, you know, some metabolites you can guess. As I said, some metabolites you can guess because based on the your in silico studies, or even to isolate the through proteomics, sorry, not proteomics, to the mass spectrometric studies, right? You can do, once you put the compound, you take the in liver microsome and then you see the mass profiling, so you can guess the metabolites. But sometimes what happens, further degradation happens in body which we have no clue about.
Okay.
And this is why sometimes what happen, actually many drug discovery projects get halted as well, because they do not really able to identify a thing which is causing the toxicity or some other thing. They can have some educated understanding from the metabolite or for the compound itself, but sometimes it becomes beyond understanding. So I do not know exactly what compound, but I think sometimes it becomes even difficult to understand as well. It could be some metabolite which you have no idea about.
Okay, okay, fine. Okay. So I have two more questions.
Yes.
Well, now in India, we have a lot of resources, for example, small bioactive molecules, compounds, everything. But none of them are really gone to from bench level to clinical. That means like at least an adjuvant therapy, like combination therapy. Either well, there is a one way to do that is in vivo model, like in humans or animal studies. Apart from that, you mentioned something about a virtual screening. Is it possible?
Yeah.
Can we check some molecule just to have some idea? Okay, this molecule can be useful for adjuvant, something like that. Do you have any suggestion on that one?
So basically, what you are trying to say that why in India we do not have many drugs in the market? Is
Yes,
that
sir, yes, sir. Because now or not every research is progressing to the clinical one. We have so much of research coming every year. And but the thing is, I like to know either any way like a virtual screen or in silico model that we can find, okay, this molecule could be useful for adjuvant or clinical combination therapy with some
right?
FDA. What is it?
I got it. Actually, the point is, we do
Uh-huh.
not know what happens in the inner story. So let me tell you.
Mm.
So drug discovery and to causing a clinical trials to take it to that level, it's a huge, humongous process where billions of dollars are invested, right? As I told in the beginning. So what
Okay.
happens is small companies, small biotech companies or small these companies, they do not have that much money to take the compound to the clinical trial level. So
Okay.
even if some company gets a compound to the preclinical development level, what they do, they prefer to sell it out to the bigger pharmaceutical companies. And
Okay.
this big pharmaceutical company take over the compound from there and then they do clinical trial. So you never
Mm.
actually get to know
Okay.
sometimes
Okay.
that
Okay.
what
Okay.
actually small companies contribution is.
Mm.
So in fact, the compounds which I showed you, which was developed in Max Planck, it was also given over as I remember when I was leaving, there was a talk with Bayer was going on to sell it
Mm.
to the buyer, that
Mm.
compound,
Mm.
right?
Mm.
The
Mm.
second thing is in India, nowadays still that R&D investment is not so high.
Okay.
In
Okay.
India, if you see mostly the companies are, either they are CRO companies, which is the contract research organization, or they are generic companies. So among the
Mm.
generic companies, there is a Glenmark, there is Dr. Reddy's, and so they are
Yeah.
launching the
Yeah.
drugs.
Yeah.
Recently, actually, Glenmark, I think, launched one drug. I forgot the name of that.
Mhm.
But then they are not really, they are mostly generic drugs. They are not absolutely novel drugs.
Okay. Okay.
So
Okay.
the investment and all these things are coming into the picture. But as you correctly said, probably this virtual and all other screening coming into the picture, the
Mm.
computer-aided drug design and taking to the
Yeah. 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 Yeah.
all
Yeah. Yeah.
together. Probably when the cost will drop down, probably all these small companies are also coming in. But I can tell you in India, the scenario is very rapidly changing and many small biotech companies are also coming up, especially in Bangalore and Hyderabad.
Okay.
Yeah.
Okay, okay. So one more last question. Well, I'm like to study some circulating biomarkers.
Okay.
Is it possible to target those things because they're all already in the blood? And is it possible how to do that? Circulating. I'm interested to target circulating proteins, for example. Okay, normally when you injected drugs, normally they're intracutaneous. The drugs will pass through the all the thing, then they will reach to the particular tissues or something where the target of protein or whatever things are residing. Well, now the new concept of liquid biopsy, like the protein itself is circulating in the bloodstream. So that's also as a marker for some cancer. So I'd like to know if it's possible to target those circulating protein biomarkers.
I think it should be possible with the new approaches coming in, but I need to be honest here that I have limited knowledge in this thing which you have asked for, right?
Okay,
So I
okay,
need to
okay.
go back to the table and I need to read
Fine,
myself about it.
fine,
Okay.
fine, fine.
So I'm sorry, I do not have a concrete answer to this question.
Okay, okay, fine, sir. Maybe shall I mail you, sir, if you don't mind?
Yeah. That would be great. You kindly email me and then I'll check that and I'll try to respond to you. Okay.
Oh, great, sir. Thank you so much.
Okay.
In
Thank
the
you, sir.
interest of time, just the last question, maybe by Sabarano Baral. Maybe you can ask a quick question, please. Then we have moved to the next
Hello.
session.
Yeah, yeah.
Am
Yes,
I
please.
audible?
Yeah, please. You are audible.
Yes. Sir, can we actually measure the toxicity or the side effects to the computational level for a drug discovery?
Absolutely, it's possible. As I have told that these days in silico studies are coming very powerful. In fact, in the compound, the last thing which I have showed, I showed you that how the benzylic CH position was replaced with benzylic CF, and the excretion rate from the mouse liver microbiome was drastically reduced, right? Actually, this was based on the prediction. So first, the in silico modeling was done. In silico, we found out that that is a hotspot, metabolic hotspot. So, and based on that only, that compound was generated. So absolutely it is done. So everything nowadays, people, before they start in vivo admit toxicological profiling, they first do in silico nowadays. So it's absolutely possible.
Okay, sir.
Okay.
Thank you.
Thank you, Dr. Sandeep. So, if you want to interact with Dr. Sandeep, you please write an email to him, and you can proceed with further discussion there. So, in the interest of time, we are moving to the next session. So, let me thank again Dr. Sandeep for a wonderful talk. And now I would like to hand over to Rajveer for further proceedings.
Okay, thank you, Dr. Pankaj. So I'm stopping my video, and you can proceed
Thank
with the
you.
next. Thank you.
Thank you, sir. Good morning, everyone. It's a great pleasure for me to introduce our next speaker for today's talk, Dr. Priyanka Singh. Dr. Priyanka Singh did her PhD with Dr. Karl Hans in the field of cancer biology from Münster University, Germany in 2011. Thereafter, she received EMBO long-term fellowship, which allowed her to pursue postdoctoral research at Biozentrum, Switzerland. Her work in Biozentrum involved understanding the mechanisms underlying brain development using fruit fly as a model organism. Subsequently, she did a second postdoc at Max Planck Institute of Molecular Physiology, Dortmund, Germany, with yet another prestigious Alexander von Humboldt Fellowship. During her stay at the Max Planck Institute, she focused on elucidating the mechanisms employed by molecular players during cell division. Since May 2017, she has been working as an assistant professor in the Department of Bioscience and Bioengineering at IIT Jodhpur. Her research group at IIT Jodhpur is taking a holistic approach to establish prognostic and diagnostic values of centrosomes in human disease and extrapolate this understanding towards identifying new therapeutic drugs. She has also received several prestigious awards as an independent researcher, which include Early Career Research Award in 2018, Innovative Young Biotechnologist Award 2019, and New Young Scientist Award recently this year by different funding agencies from India. So, without any further delay, please join me in welcoming Dr. Priyanka Singh to address the session on tools and techniques in proteomics. Thank you, ma'am. Over to you. Looking for an exciting session.
Thank you, Rajvi, for the nice introduction. So I hope my slides are visible. Can somebody confirm that?
Yes, ma'am. Your slides are visible and you are audible.
And if I'm moving to the next slide, it is also changing.
Yes, ma'am.
Okay. So I welcome you all. We had a very nice session in the morning about the drug discovery. So I'll now take you to the tools and techniques which are used in the field of proteomics. So the talking about the omics cascade. So omics is a suffix that is kind of used to indicate large scale data or information which is used to understand the life. So the information in life sciences is carried from DNA towards the protein level. So the information which is at the DNA level is translated, it's transcribed to RNA, and the information that is present in the RNA is then translated to form proteins. And then we have body basically breaks down these food, drugs, or chemicals that we consume or so into small molecules which are called metabolites. So you can collect a lot of information about the living system by collecting these data that is present at different levels. So if you're looking into DNA sequences, then in the omics cascade, the term that is given to it is called genomics. If you're looking at the information large, or collecting the large amount of information at the RNA level, then it's called transcriptomics. If you're understanding the proteome or the protein in a living system, then this is called proteomics. And if you're trying to look into the metabolites under certain set of conditions in different individuals, then this is termed as metabolomics. So our focus here will be on proteomics. So before we dig deep into the tools and techniques of proteomics, I'd like you to brush up your understanding about proteins, because this is very important to develop any kind of tools. You should understand the system, how it works, what is the chemistry behind it. So proteins are basically made up of amino acids. So amino acid has a amino group, it has a carboxyl group, and then there is a alpha carbon to which the hydrogen atom is attached, and there is a R group for the side chain. So there are around 20 naturally occurring amino acid. They have different types of R groups, each one of them, which gives them a distinct property. For instance, these amino acids are categorized as polar, non-polar amino acid, positively charged amino acid, negatively charged amino acid, based on the side chain that they have. So these amino acids, they come together, so they polymerize and they form this polypeptide chain, which forms the protein. So the two amino acid, what happens is that the carboxyl group of the first amino acid, it joins with the amino group, the next amino acid, and it forms a kind of peptide, a bond, which is called the peptide bond. So the polypeptide chain, it has a sort of polarity. It has an amino group where you find the free amino group, and then there's a carboxyl end, where you find the free carboxyl group. The peptide bond is very, it's rigid. It's a CN bond. It has a double bond nature. So the rotation is restricted around the peptide bond, but the rotation around the single bond is possible. So this property of the kind of amino acid that are present in a sequence basically plays an important role in giving a structure to your protein. So protein structures have been classified into at four hierarchical levels, starting from primary structure, then to secondary structure, tertiary, and quaternary structures. So primary structure is nothing but it is just the sequence of amino acids which are present in your proteins. Now this sequence of amino acid basically will allow this chain to fold into some certain patterns, and that will form part of, or that will result into formation of secondary structures. There are two major secondary structures present in proteins. One is alpha helix, which is depicted in this diagram here on right side as this green helix. The other kind of secondary structures are the beta pleated sheets, which are represented as arrows, these blue arrows going back and forth in this chain. So these are the major kind of secondary structure. These are regular substructures which you find in your proteins. Now these substructures, they can again fold and come close to each other with the help of some weak interactions, and they form this three-dimensional form of a protein, which is called the tertiary structure. Some proteins only have tertiary structures, whereas the other proteins, they also, you will see quaternary structures in those proteins. So these are complex structures. For instance, in hemoglobin, you have four different polypeptide, they fold individually and they come close to each other and they form these quaternary structures. And this hemoglobin helps in the transport of oxygen. Now, we need to also understand what are the forces which are present in your protein, which help in folding of this protein, help in stabilization of the protein. Now, why this is important? Because in any tool and technique, if you want to purify your protein for proteomic studies, you would want to play around with these forces only. So there are covalent forces are of course required for forming the polypeptide chain, but there are several weak forces also which play an important role. One such weak force is the hydrogen bonding. So hydrogen bonding basically involves the hydrogen atom which is attached to an electronegative atom, and it can form a sort of weak bond with another electronegative atom. So this hydrogen bonds is also required for the secondary structures that we see, alpha helical or beta pleated sheaths. They are stabilized by the hydrogen bonds. We also have sometimes in protein structures, we find this metal ions. For instance, I took the example of hemoglobin. It has this ferrous ions or so. It forms coordinate bonds with surrounding residues in the protein, and that also helps in keeping the structure together. The hydrophobic interactions are the weak interactions or the non-covalent interactions that plays an important role in the stabilization of the protein. So this will involve hydrophobic residues. For instance, in this diagram, you can see leucine, valine, isoleucine marked here. So these are hydrophobic amino acid. Hydrophobic meaning that they will repel water. They don't like being surrounded by water. So they try to be present with the inside a fold so that they can avoid contacts with the water molecule, which is on the outer side of the 3D structure. Then there are disulfide bonds. These are covalent bonds basically that are also very important and frequently found in a lot of proteins. So disulfide bond basically result because of certain amino acid like cysteine. It has a sulfhydryl group, SH. So if you have two cysteine molecules in close vicinity or so, those will basically get reduced and you will have the disulfide bond formation. Then there are electrostatic attractions, which are also weak attractions. For instance, you have two amino acids in close vicinity. One is positively charged amino acid, the other one is negatively charged. So because of that electrostatic interaction, that will also help in keeping this structure together. And this understanding of basic knowledge is what has been exploited in a lot of techniques to purify these proteins and to develop these techniques around this idea. Now, understanding the solubility of protein is also very important. So I'm not going to go into too much depth about this, but just to remind you that you should also understand the charge on a protein. So proteins are charged molecule. As I said, there are positively charged amino acid, negatively charged amino acid. So in a protein, you will have all kinds of amino acid. So overall, they will contribute to the charge of the protein, and also the kind of the solution or the pH of the solution in which the protein is present, that will also influence the charge. So in order to understand protein solubility, let us look at this isoelectric point. So in a more simplistic view, if we look at a particular amino acid, which is denoted in this diagram in a very general way, if you're working at a lower pH, basically, so here you have more protons. So then your amino group is fully protonated, your carboxyl group is fully protonated. So here in this equation, you see NH3 plus COOH. Now, when you start titrating this amino acid, you add some sort of base to your solution. Then the protons will be donated by the amino acid to neutralize that base. So first the carboxyl group starts giving the proton. So now you will have this kind of species being formed, which is called the zwitterion, where you have amino group which is protonated, NH3 plus, and the carboxyl group COO minus, which is in a deprotonated form. So in this form, your net charge on the amino acid is 0. You have equal number of positive and negative charges. So this form is called the zwitterion form, and the pH at which this happens is called the isoelectric point, or the PI. So there is no net charge on your, if we extrapolate this understanding to a protein level, then on your protein there will be no net charge. Now, if you're further titrating it, then again, finally your amino group, it will start giving out the proton, and finally you will have this species where your carboxyl group is deprotonated, your amino group is deprotonated. So in this form, your amino acid is has a net negative charge. So you have to have an understanding that the pH also influences, or pH of your solution influences the charge on your protein. So if your pH is less than the PI, isoelectric point, overall your protein will have a net positive charge. If your pH is more than the isoelectric point, then protein will have an overall negative charge. And this is an important understanding that is also being used in some chromatography techniques, which we are going to discuss in the later slides. So why do we need proteomics? Because we have a lot of, we have genomics, transcriptomics, then why people decided to actually also look into proteomics. Now, understanding proteomics, or to spend time in looking into proteins, their structure, function is very important in order to understand life, because the level of transcription of gene, so the number of transcripts that are being formed, it's not equal to the level of gene expression. Does not necessarily mean that they're going to be expressed and they're going to result into the formation of protein. So if you actually want to understand the functional level of understanding of any system, you would like to look into the final product of this, the flow of information that you have. So protein is at the final end. So this is what people are trying to now spend time in understanding the proteomics of an organism under a set of different conditions. So another point that you should also keep in mind, that the information that is stored in genome is also expressed differently in different cell types. So if you are trying to understand different cells, or for instance, in some certain diseases, only certain tissues being affected. So understanding genomics will not give you too much information, but you have to, at the end, look into the protein level expression in these tissues also. So in this, you have to keep in mind that one gene can result into many transcripts. One transcript can result into many proteins. So if you would like to have an understanding of the functional level understanding, then this is your target. You have to look into the protein functioning, its structures, its organization. And one protein can also basically be involved in many processes also. So that's still not the end of the story. You also understand if a protein has a particular one, more than one also. So what is proteomics? So here the stage to now move forward and understand exactly what is proteomics. So proteomics is the large scale study of the proteome. So what is proteome? It is the set of proteins which are produced in an organism or in certain biological context. So you are basically looking into the entire proteome, large scale analysis of the entire proteome in an organism. But proteomics studies also quite challenging, because as I mentioned in my previous slide, that proteome is not constant. It differs from cell to cell and also from time. So the proteome that you have at this age might be different from the proteome that you will have when you are 60 years old. The proteome also differs, like if you have a disease, healthy proteome is different from diseased proteome. And the environment also play an important role in regulating a lot of factors, the way your proteins are being expressed. So that will also contribute to it. So understanding or the information that is revealed by proteomics is at a lot of levels. For instance, you can look into the localization patterns of your protein. So you can look into when and where your proteins are being expressed. You can look into the dynamics of your protein. So basically the rate at which these proteins are being produced, they are degraded, their steady state abundance, or their movement from one subcellular compartment to the other. You will look into the modification and the regulation of these proteins. So basically you can look into how these proteins are modified. Modified meaning that there are certain post-translation modifications, example phosphorylation. Certain proteins are phosphorylated at a certain time in the life of an organism, or if we're talking at the cellular level, at the life of at a certain cell cycle event. So post-translational modification is also one field where the proteomics is developing. Then functionality. So basically, the involvement of proteins in the metabolic pathways, and then the protein network. So how these proteins interact with one another. So all these level of information can be revealed by techniques which are used to do proteomics analysis. So there could be different types of proteomics that you can do depending upon what you want to learn from the living system. You can do structural proteomics. Structural proteomics meaning that you can kind of look into the structure of the proteins, or you can look into the so you are trying to understand the protein complexes present. You can look into the organelle compositions, for instance, particular organelle in my body, what are the proteins that it is made up of. You can do functional proteomics, where you are looking into the functioning of a protein. So a lot of techniques are actually available to do functional proteomics. For instance, you can do affinity purifications combined with some pull downs of the protein complexes. You can do knockout approaches, wherein you can knock out certain genes which are expressing for that particular protein, and then you can see how that is affecting the overall functioning of the organism. Yeast genomics is actually quite a lot used, not just for genomics level, but also for proteomics levels understanding as well. But yeast is a very, it's a single cell organism, so it is a widely used system in life sciences. So it is easy to manipulate the gene of in the yeast. So for instance, these cyclin CDK, which are involved in the cell cycle events, they were identified by manipulating the or by mutating certain regions in the genome of the yeast which code for these proteins, and then this allows them to actually do a high throughput kind of screening, where they could identify that this particular gene is essential, is coding for this CDK cyclins complexes, which is required for cell cycle events for mitosis also. Then you can do protein expression profilings. So you can so that basically has an application, for instance, in disease mechanisms or so. So you can look into the expression profiling in the healthy patients, in the diseased patient, and this will basically give you an idea of what kind of proteins are being upregulated, downregulated in the diseased patients, and what kind of pathways are being affected or not. Then you can kind of look into the post-translational modifications on your proteins. These post-translational modifications are very important for certain biological events. For instance, phosphorylation, proteolysis, glycosylation, they play a very important role in cell signaling, which is also part of etiology, which is basically could contribute to a etiology of different kind of diseases. Then you can look into protein-protein interactions. So there are different tools available for protein-protein interactions. For instance, there is yeast-2 hybrid screening, co-immunoprecipitation, FAST libraries, which will tell you which is the what kind of proteins your protein of interest is interacting with. So if in a disease condition that protein is misregulated, then exactly what kind of proteins or interactions are affected, and you can anticipate what kind of pathways will get affected by that. So, so as I said, there are actually different kinds of proteomics. So, depending upon what you want to learn. So, majorly or broadly, we can categorize them into expression proteomics, structural proteomics, and the functional proteomics. So, there are different techniques one can kind of use. A combination of techniques also are there, where you can do expression profiling, you can look into the structural analysis of your proteins, you can look into the functional aspects of your protein. So these are just few of these tools that I've mentioned. It's not possible for me to discuss all the tools in today's lecture, but I'll try to cover as much as possible. So 2D gel electrophoresis and mass spectrometry are used for expression proteomics. For structural proteomics, techniques like X-ray crystallography, NMR, some predictive modeling tools are used to predict the structure of your proteins. Then functional proteomics, you use techniques like yeast to hybrid assays to understand what are the protein interacting partners. You use ELISA or protein microarrays, which are the high throughput screening approaches to look into the functionality of your proteome. Then, so proteomics basically requires a sort of workflow that you have to take. First, you have a sort of sample from which you would like to extract out your protein. So after extraction, you will have a protein in a sort of soluble form. It's up to you now how you would what you want to do with your protein. So basically, you can, if you want to do protein interactions, or if you specifically know which kind of protein you would like to study, then you can do immunoprecipitation to precipitate that particular specific protein. You can you also do size exclusion chromatographies, wherein you can kind of isolate that particular protein of interest from the rest of the protein. You can do STS stage analysis. It is an analytical kind of a technique where you can confirm that your immunoprecipitation size exclusion column, whatever elevate you have as your protein of interest. Once you have a protein of interest in your hand, you digest it to make smaller peptides or so. Now these peptides, if you want to look into post-translation modifications, then you have to enrich those peptide for the post-translation modification. So you use certain techniques to enrich the, for instance, if you would like to enrich some phosphates, peptides which have phosphates or so, then you use some kind of technique where your phosphate containing peptides are enriched. So basically some sort of affinity chromatography techniques is being used here. And then these peptides are fractionated and they are separated, and then you put them into these mass spectrophotometers, which will give you sort of mass to charge ratio with which you can kind of again put this into a sort of databases where it will give you the identity of that particular peptide, and from that you can basically tell which kind of protein it was. So every step is very or the techniques which are used at every level, extraction, the digestion, separation, it actually utilizes the understanding of protein chemistry. So let us look into protein extraction methods. So whenever you pick up any paper to read about proteomics, you will realize that every paper uses a different kind of technique to extract your sample. Now this is because of the type, because you need to understand what kind of sample you have. You cannot basically have one protocol and apply it to every kind of sample. So there could be two types of sample broadly that you are dealing with. Either it's a solid sample, for instance, it's a tissue, it's a plant, sort of part of a plant from which you would like to extract the protein, or it could be a liquid sample, for instance, some body fluid, serum, or so. So you need to process it differently depending upon what kind of protein you want to extract, from exactly where the protein is localized in your sample or so. So if it's a solid sample, the first step is definitely to homogenize your sample. So you use some mechanical methods. For instance, you use a blender, you use either a mortar pestle or so to homogenize the sample. And then you're going to lyse your sample. So lysing can be broadly two types. Either you are using a physical method where you're using certain heating method or so, or you're using a sonication method. It can be a chemical method where you add some detergents. So detergents will basically lyse the membrane which is present in your sample. And if a protein is in the cytoplasm, for instance, then it will basically come into a solution. Or you can use some cheotropic reagents. For instance, urea, guanidinium hydrochloride are being used. So these cheotropic reagents basically play around with these weak interactions that you have in your protein structure, and that will help you to in the lysis step as well as in your protein purification steps as well. If you have a liquid sample, on the other hand, then you can kind of isolate the cells which are expressing your protein of interest. So for that, you can use some sort of centrifugation techniques. For instance, density gradient centrifugations are highly used. You prepare a gradient of certain inert agent, for instance, cesium chloride or sucrose gradients are mostly used. So then you load your sample on top of this gradient and you centrifuge your sample. So then your sample will start passing through this gradient, and the place at which the density of your cells or your is actually equal to the density of that gradient, then this is where it will settle. And then you collect these fractions, and then you analyze in which fraction my protein of interest is going to be, and you deal with or you start processing that particular fraction or so. So again, then this is the same also. Once you know that this is the fraction in which I have a cell which is expressing a protein, then you lyse that particular fraction by using the similar kind of physical chemical methods. Then you so finally you will have a soluble protein after lysing. Once you have a soluble protein, you can depending upon if your protein is highly expressed, you don't need protein precipitation at certain points. But if your protein is not highly expressed, then possibly you need to add a precipitation step or so. There are different ways by which proteins can be precipitated. For instance, you can use salting out method where you add salt like ammonium sulfate. So ammonium sulfate basically reduces the solubility of your protein because it also it replaces basically the water around your protein or so. So your protein is precipitated. You can also play around with the pH. So if you're playing around the pH, then you can trichloroacetic acid is also used in that. So that basically influences the pH, and that also results into the protein precipitation. But for that, you should be aware of the isoelectric point of your protein of interest. You can also use some polymers or some organic solvents, which basically plays around with the, which also basically help in the protein precipitation, and they will help in your precipitating a protein from the solution, and then you can kind of isolate that fraction, and you can process it towards purification steps also. So what I want you to get from this, that the isolation techniques or the extraction techniques of your protein should be chosen with the method of analysis. So later stages, what you want to do with that protein, that you should always keep in mind. Because for instance, if you would like to do a functional analysis, you don't want your protein to be denatured. Denaturing meaning unfolding of the protein. You would like to keep it in a folded state. Then you would like to avoid certain chemicals which can result into denaturation of the protein. For instance, some coechotropic agents that I said that result into denaturation of the proteins. So then you would want to avoid those. So every different papers, if you take, which are used in the proteomics, where they have extracted the protein out of an organism or biological system, you will see different papers will use different protocols for protein extraction. So you need to basically take time to understand that why this kind of method is being used. So people use sometimes combinations of these this learning or this tools in order to have their final protein in their hand. Now, protein purification step that I mentioned later on, that you can do by different ways. One very widely used technique is called chromatography. So chromatography basically it's an analytical technique which is used to separate mixture of compounds, which can be further used for analysis. Chromatography has two phases. One is mobile phase, the other one is called the stationary phase. So mobile phase is the moving phase. And the stationary phase is the non-moving phase also. So basically here, below the two diagrams, what you see on the left hand side, it's paper chromatography. So in paper chromatography, your stationary phase is going to be the paper, and mobile phase is the solvent, which is moving through capillary action in the upward direction in the paper. So depending upon what is the solubility of your analyte to the solvent and how well it is absorbed to the stationary phase, your analyte will start moving. So so basically, if you have a mixture of these analyte, then they will be separated as your solvent will move in the upward direction in this paper chromatography. So more soluble a compound is in the mobile phase, more it will move along the mobile phase. In the stationary phase, if a compound is attracted or absorbed, then this will drag the movement of that compound. So this is one type of chromatography. Another version of this chromatography, this is column chromatography. Here you are using a column. So in the column, you will have it is kind of packed with a stationary phase, and then you have a mobile phase moving along the column. So whatever your solvent that you add to it. This is a very conventional and very simple to use way of using column chromatography. Here is a clamp with which you have a support to the column. Below is a flask where you're collecting your sample. So you can wash this, you add a mobile phase, it is getting collected in this flask, and then you add your protein mixtures on top of it, and that protein is going to be separated. Now separation of the protein will depend upon, so you can have variations in the column chromatography, and you can separate it on basis of different criteria. But before that, there are actually basic steps that you follow with column chromatography. First of all, you have a sort of binding buffer that you pass through your column. So your beads or whatever the stationary phase that you're using in your column, it is equilibrated with that binding buffer. Then you add your protein of interest in the binding buffer. So your protein of interest will have affinity towards the stationary phase. It is going to get attached or the movement will be slow of that particular protein of interest, whereas the rest of the protein will move faster. So they will move first out of the column. Then you wash. You also add washing buffer through the column. So your non-specific proteins which you don't want to collect, they will be coming out of the column. And finally, you will add elution buffer, which will help you to elute your protein of interest from your column. So nowadays, your column chromatographies are attached to these detectors with which you can actually, or spectrophotometers with which you can actually look at the elution profile. So this is the elution profile for column chromatography. This here is the absorbance on the y-axis, and on the x-axis is the column volume. So when you equilibrate your column, then this is so absorbance basically, because we are working with protein, majority of the times we use 280 nanometer absorbance. If you have a fluorescent tag, you can actually use a specific wavelength of light to detect that particular elution profile. You basically, when you apply your sample, you will have a rise in the absorbance. This is because when you add your sample, proteins are basically flowing through the column and coming out. So this is all non-specific. This bigger shoulder kind of thing that you see here is all non-specific things which are coming out of your column. And here then you wash the column further. 2 to 5 column volumes are used to wash the column. And then you add your elution buffer, and then you will have a peak here. That peak basically is your specific protein of interest that has come out of the column after adding the elution buffer. So you can collect this particular fraction, and then you can analyze it and use it for your further work. So as I mentioned, there are different types of column chromatography. You can exploit different properties of a protein, and you can design different types of chromatography. One is called the affinity chromatography. So affinity chromatography basically uses affinity of the stationary phase towards your protein of interest. So this is a very simplistic diagram representing it. So your matrix has a specific ligand, and your target protein has a sort of and this ligand has a sort of affinity towards your target protein. So when you add your protein mixture onto affinity column, only your target protein will bind to it, and rest of the proteins will start flowing down as you start washing your column. Then eventually, if you want to now collect your target protein, then you add a sort of competitor which will bind to that ligand, and your protein of interest will come out of the column. So there are different, you might have heard about a different type of columns are available. For example, there is nickel NTA column. Nickel NTA column is used for the for uh, purifying proteins which have histidine tags. Then you have glutathione columns with for proteins which has GST tags. So there are different types of ligand and receptor kind of combinations that you can exploit in this. Then the other one is called the ion exchange chromatography. So ion exchange chromatography basically exploits the charge on your protein. So you can have column which can be either positively charged or it can be a negatively charged. So if it is a positively charged column, it will bind to proteins which have a negative charge. And if it is a negatively charged column, it is going to bind to the proteins which have a positive charge. So for this, you need to understand that at what pH my protein will have what kind of charge. For instance, so you need to understand the isoelectric point of your protein of interest. For instance, I know that my isoelectric point of my protein is 6. So if I'm using a pH of 8, so basically at that pH, my protein is going to be, it is going to have a net negative charge. So in order to isolate that protein, I would like to use a column that has a positive charge. So again, the steps remains the same. You load your protein, your protein will bind to your column, rest of the protein will start floating. So you should also understand when you're working with protein, a mixture of proteins, it's not just your protein of interest that will have the opposite charge. Maybe there are some non-specific protein that will also bind to it, to that column. Later, then you elute those proteins and collect those proteins. You analyze which fractions have my protein. You can check the this elution profile also. You can do a STS page analysis to exactly look at the size of your protein. That this is the specific size I have for my protein of interest. But as I said, you can imagine that this after this technique also you will have some non-specific proteins that are coming out with your protein of interest. So at times what people do is that they combine 2 or 3 such kind of chromatography technique. For instance, what one can do is that you can do first the affinity chromatography step, wherein your protein first, if your protein has a histidine tag, then you can purify his containing protein only, and then you put it onto an ion exchange chromatography, and so you will reduce the non-specific contaminants which are in your purification steps. The third kind of column chromatography, which is also widely used, is called the size exclusion chromatography. So this basically separates proteins based on their size. So here your stationary phase is basically named in such a way that when you add a protein mixture in it, your small proteins will enter the matrix, whereas the large protein will actually move around the matrix in the void volume. So your larger proteins will going to be eluted first because they have less sort of space that they're traveling in the column. Then your medium-sized protein is going to be eluted, and then your small proteins are going to be eluted. So you can combine certain techniques depending upon exactly what kind of sample you have and what level of impurities you have and how you would like to isolate those impurities from your protein of interest. So nowadays, these column chromatographies are automated. We use these machines which have automated they have this system wherein you can actually load your sample, and then they will take up all the washing solution and elevate solutions or so. You have to just fit in all the make that sort of program in your computer. So we use so these are called FPLC, which is fast protein liquid chromatography. So these are medium pressure chromatography technique in which you use pump to control the speed at which the mobile phase is traveling through the stationary phase. Because these are all pre-packed column, they can actually, when you pump some certain solutions through the column, so they can basically survive certain amount of pressure. So that is kind of being taken care of by the sort of software that you have in your FPLC. So here is the flow path of a typical FPLC, what you have. So basically it usually has this kind of pump. So pump will have a number of inlets. For instance, in this case, you have 2 sort of inlets. Then you have the sort of sample injection valve or so. And then you have UV detector. So UV detector will be there. So any sample that is passing through the UV detector, you can basically use the check. You can basically look at the absorbance at a number of wavelengths in this UV detector. Then you have a conductivity meter, which will help you to keep a note as to what is the pressure that is in your system, because your column can, as I said, column can survive certain amount of pressure, because if you apply too much pressure, your stationary phase will break and it will be destroyed. So then you have to always keep a sort of alarm that if whenever my pressure is above this particular limit, then the flow should stop. Then there is a fraction collector wherein you have certain plate where you can decide how much volume will be collected in each well of the plate. And you have some waste collections also. So after your protein purification, as I said, you need to analyze also that what is my protein of interest, what is the size of my protein of interest. And if you're actually aware of the size, then you need to still analytically verify that. So you use electrophoresis technique for that. One widely used electrophoresis technique in protein chemistry is called the STS page. STS is a name of a detergent, sodium dodecyl sulfate. Page stands for polyacrylamide gel electrophoresis. So electrophoresis basically means in general in electrophoresis technique, you have an electric current. Basically, you have a negative electrode and a positive electrode, and you have this sort of buffer system, and you have a gel. So a sample is loaded onto this gel, and your sample is moving under the influence of this electric current in one particular direction. So your separation in this kind of electrophoresis technique usually is on the basis of either mass, the charge, and as well as the shape of the protein. So in STS stage, what is being done is that the charge and the shape factor are being negated because you use STS. So STS, what it is doing is that STS is it has a negative charge. So it basically wraps around your proteins. So overall, it gives a negative charge to the protein. So your proteins usually, depending upon the kind of amino acid it is made up of, it can either be positively or negatively charged. So the movement in the electrophoresis will be also based on charge if you don't STS. But because you are adding STS, you have negated that inherent charge in your proteins, and now your proteins will have negative charge, which is proportional to the polypeptide chain length. You add a certain reducing agent like beta-mercaptoethanol in your solution. So basically, this beta-mercaptoethanol, what it is doing, it is breaking up these disulfide linkages. So that basically result into denaturation of the protein. Your protein is no longer in a folded state; it is in an unfolded state. So now the shape has been negated, the charge has been negated when you prepare a sample like that. So now the movement of your polypeptide or your proteins is based on only their size, molecular weight or so. So usually you add a sort of ladder or a standard along with your sample. So the standard of the ladder basically is comprising of a collection of bands of known sizes. So you can kind of compare your unknown protein band with reference to that ladder lane, and you can kind of predict what my protein size would be. Then this electrophoresis the other kind of electrophoresis technique which is also used quite a lot in proteomics is called the two-dimensional or the isoelectric focusing. So in isoelectric focusing, basically it exploits two properties. First is the this isoelectric point of your protein of interest. The other one is the molecular size of your protein of interest. So what you do first in this 2D electrophoresis is that you load the mixture of your protein onto a isoelectric focusing tube gel. So you have this kind of a tube gel which has a range of pH from 4 to 10, from starting from one end of the gel towards the other extreme end of the gel. So when you load your mixture of protein and then you do electrophoresis, then your protein is going to move along this pH, but the pH at which it matches the isoelectric point of your protein, then there will be net zero charge. So your protein will no longer move. So it will kind of settle at a particular sort of position in this isoelectric focusing gel tube. Then you take this gel tube, you put it on top of a STS page, and then you again do a sort of electrophoresis. So now each sort of band which was there, which was at a particular PI, it is going to now move on the basis of their molecular weight. So each band, so this is how this is a picture of the left hand side you see. This is a gel, how it looks like after isoelectric focusing. So after, so this is a Comasi stain gel. So you use a Comasi staining dye to actually visualize the protein bands. So for instance, if you compare this band A with band B, so band A here will basically has a protein that has a PI around 5.5 and it has a higher molecular weight, whereas band B has a higher PI and lower molecular weight. So basically this kind of technique will help, because in your protein mixture it can often happen that you will have a protein which have same kind of molecular weight. So if you do only STS page, then those two proteins will end up in a same band. You will not be able to separate them. But if you combine it with isoelectric focusing, then it's quite rare that the two proteins will have also the same PI. So then at least at one level, there will be separation between these proteins. So this technique provides a better sort of separation between the proteins from in a mixture. So these bands are later then cut and they are sent for mass spec analysis, and then you can also, if it's the unknown proteins, you can kind of look into the identity of these proteins. Now, often for function analysis, Western blotting technique is followed after STS page. So for Western blotting, basically you need to have antibody against your protein of interest. So what happens in Western blotting is that when you have your STS page where you have separated your protein, then you prepare kind of a sandwich of your gel along with the membrane on which you would like to transfer that particular protein. You use a membrane like PVDF or nitrocellulose membrane, and then you put it into this sort of tank, which is again a sort of has these electrodes from minus to plus, and the sandwich is arranged in such a way that your protein will move from your gel towards the membrane. So once your proteins are transferred to the membrane, then you can kind of process this membrane. You block this membrane with certain BSE or milk or so. Then you provide primary antibody against your protein of interest. That antibody will basically bind to the protein of interest on your membrane. And then you can provide a secondary antibody. Secondary antibody has affinity towards the to the primary antibody, and it is also conjugated with an enzyme. Widely used enzyme in the Western blotting has this HRP enzyme, phosphate peroxidase enzyme. So that enzyme is basically being used to so you provide a sort of developing substrate to this reaction. So your substrate is going to be converted into the product, and a light is being also released in this process. So it's a chemiluminescence phenomenon which exploits this HRP enzyme. So HRP converts this substrate into a product, and this results into luminescence. So then this light is kind of red by this autoradiograms that you have. So then in your membrane, you will find bands only for your protein of interest, and the rest of the bands will not be visible. So this will help you to detect specifically or to basically further validate that the protein that you've identified is indeed a protein of interest, because you are using a very specific antibody against that protein. Then there are other assays which are designed, which are antibody-based assays which are designed. So this Western blotting utilize antibodies. And there is ELISA. So ELISA is also sort of, you can say it's a sort of high throughput sort of assay where usually you use this 96 kind of well plate. On the well of each well, you are basically prepare a coating of antibody which is against your protein of interest or your target protein. Then you provide this mixture of proteins onto that onto each well. So your protein will bind to the antibody. Then you provide a secondary antibody against your protein of interest, which is conjugated to the enzyme. And then you provide a substrate which is going to get converted into a product, and in this process it will give out light, and you can measure that absorption. So this so there are different versions of ELISAs also: direct ELISA, indirect ELISA, sandwich ELISA. So I will not go into too much depth around it, but these are certain tweaks around the basic idea. So this is a sort of diagnostic tool that is being used. For instance, if you would like to identify if a sample has a viral antigen or not, then you can basically do this diagnosis using ELISA. You can coat your well with the antibody against that particular viral antigen, and then you provide that sample or the body fluid in your plate, and then if it will have an antigen, then it will bind to the antibody, and the secondary antibody will also bind to it, and this will give you a signal. But if there is no antigen, then your secondary antibody will not bind, you will not have any signal. So another version of ELISA, basically it's called protein microarrays. So it is just like DNA microarrays, where people are trying to make these chips, protein chips basically, where the solid phase is basically coated with this library of proteins, and then you can utilize these to look into the protein-protein interaction, and you can look into the functional proteomics also. Now, another method that is also used is called immunoprecipitation. So, immunoprecipitation will help you to look into precipitate your interacting partners. It is a technique widely used in functional proteomics. So in this case, immunoprecipitation is usually carried out at a very small scale in these small tubes or so. So what you have usually is these kind of beads onto which your antibody, primary antibody is being, not primary antibody, these are capturing antibodies actually, which are being conjugated to it. So what you have, you prepare your protein mixture, you mix it with your primary antibody that basically binds with your protein of interest. And then you add it onto these beads, which has a capturing antibody attached to it, which captures your primary antibody. So any protein that will bind to your protein of interest will also kind of get bind to the bead, and rest of the protein will be washed in the washing steps. And finally, you can elute your protein of interest along with the complex with which it was binding. And you can do STS page analysis, Western blotting, and others kind of analytical techniques I mentioned in order to reach to a conclusion if my protein is interacting with the other protein or not. The other kind of techniques used for functional proteomics is the yeast to hybrid system. So yeast to hybrid system is widely used for understanding protein-protein interaction. So what it does, it exploits the property of transcription factors. So transcription factors basically they have modular nature. They have two domains. One is called the DNA binding domain, depicted as DB in this diagram, and it has an activation domain, which is depicted as AD in this diagram. So the DNA binding domain of a transcription factor, it basically binds to the DNA promoter sequences next to your gene of interest. And the activation domain basically interacts with the transcription machinery, and it brings the transcription machinery to the site, and it helps in the protein, it helps in the transcription of the gene of interest, which is next to your promoter. So what researchers have done, they have basically separated this DNA binding domain and activation domain into two different cloning vectors. What you can do is you can basically, if you would like to understand the interaction between two proteins, for instance, X and Y, you can basically clone your protein X in a vector that has a DNA binding domain. You can clone your the genes for your protein Y into a prey vector which has an activation domain. And then you can co-transfect them into a one yeast cell. So if the two proteins are interacting, X and Y, they will bring the activation domain and DNA binding domain close to each other. So now this will be read as this will act as a complete sort of transcription factor, which will carry out the transcription of your reporter gene, which is downstream of your promoter region. If X and Y, they do not interact, then the activation domain, DNA binding domain will not come together, and they will be separated. So they will not carry out the transcription of the reporter genes. So it was all based on interaction between X and Y. So now you can play around and have a number of reporter genes in different systems. This is a widely exploited tool. This is also you have kits also now available for yeast to hybrid systems. So you can use a number of reporters. For instance, some of the classical used reporters are histidine 3, uracil 3. So histidine 3 codes for histidine, which is the amino acid which is required for the growth of the yeast. So what you do is you basically have a yeast which is an auxotrophic mutant. So you basically do not provide histidine in your growing media. So the cells or the yeast cells will only grow when the reporter gene is being expressed. So if the reporter gene are not expressed because the X and Y are not interacting, then the yeast will not grow. So this will be an indicator that your proteins are interacting partners or not. Similarly, uracil 3 codes for uracil, and that is also utilized as also in auxotrophic mutant as a reporter gene. The third classical reporter gene that is used is called the LAGZ reporter. So that is a sort of derivation from the lag operon. You have this these genes which code for beta galactosidase. So you provided a substrate which is converted to a product with the help of a beta galactosidase, and it will give you blue color also. So you can have a blue-white screening. So if they're interacting, they will give you blue color. If they're not interacting, then it will give you a white color colonies or so. Then moving forward to the more towards quantitative proteomics also. Sorry. So basically the sample of interest that you can have, there could be different types of samples from which you're extracting your proteins. You can run a 1D gel, which is STS page, or you can run a 2D gel. 2D gel will help you more resolution of different protein mixtures. You can excise these bands from these gels, solubilize it. You can do in gel proteolytic digestion. So your proteins which are in the gel are now converted to peptide mixture. You can basically then do a mass spec analysis of these protein mixtures. You will get these kind of mass spec outputs, which will give you the identity of the amino acids that this peptide was made up of. You do a data search with these peptide sequences, which will give you the identity of the proteins that was present in your peptide mixture. So this is one approach that is being used. The other approach is called the shotgun proteomics, wherein you skip all this middle steps of running the gel. Directly, you can solubilize your protein, and then you can do an in solution proteolytic digestion, and you get these peptide mixtures, and that can directly be processed by mass spec analysis or so. So talking about mass spec, so mass spec basically separates ion based on mass to charge ratio. So there are the three major components of a mass spec are the ion source, analyzer, and detector system also. So below you can see a simplistic representation of mass specs. So the ion source kind of produce gaseous ions. Then your analyzer basically it resolves the ions based on the mass to charge ratio. And then you have a detector that is going to detect those ions, and then it will record the relative abundance of these resolved ions. And finally, you will have this kind of mass spectra where you will have these kind of peaks, which indicates the relative abundance of these peptide with respect to the mass to charge ratio. Now, let's look at the kind of basic idea as to what are the components of mass spec. So mass spec basically involves 4 major steps. One is the ionization, the other is acceleration, then deflection and detection. So in the ionization step, what is happening is your sample, the atoms in your sample, in these atoms, the electrons are knocked out and positively charged ions are being formed. So ionization is happening here. These ions that are being formed, these are accelerated to have same kinetic energy in the second stage. Then these ions move like a beam into the chamber that has electromagnets around it, and that this is where the deflection happens. So the ions basically deflect based on their mass to charge ratio. So the lighter lighter ions, they deflect more in this electromagnetic field. And if there are more charges on the ion, then they also deflect more. So overall, it's a combination of mass to charge ratio, their deflection. Finally, these depending upon what path they are taking, they will reach the detector, and that detector will convert that signal into a sort of electric current, which is amplified, and the signal is recorded by this recorder. So let's look into each of these components into some more detail. So talking about ionization. So what is happening in ionization? So you have this vaporized sample that is entering into your ionization chamber. So the particles in your sample, which are atoms or molecules, they are bombarded by this stream of energetic electrons. So you have this electrically heated metal coil which generates electrons, and this vaporized sample will bombard these electrons. So what will happen in this situation? That your atom will basically lose one or more electrons, and they will become positive ions. So now your ion repeller will basically repel the ions from the ionization chamber towards the accelerator. Now, what is happening in accelerator? In the accelerator, you have these sort of optic plates which are arranged. So this is at a potential of around 10,000 volts or so. basically this kind of helps in forming this kind of ion beams, which enters in the deflection. Now in the deflection chamber, what is happening is you have this electromagnet, and the ion stream basically enters this electromagnetic chamber. The mass to charge ratio, basically these ions are basically sorted based on their mass to charge ratio, which is influenced by a number of factors. So it is influenced by D square. B basically stands for the strength of magnetic field. R is the radius of this deflection tube, and V is the potential. So basically, you can play around with the magnetic field, and you can kind of choose what kind of mass to charge ratio is kind of deflected in this system. So if we note in this diagram, there are in the mixed ion stream, when it enters the deflection chamber, there are these three streams which are being deflected, A, B, and C. A is denoted by this blue dotted line, B is by the green line, and C is by the red line. So the ion A, basically this means that the ion beam A, this is the most deflected, so it has the least mass to charge ratio. The C, it is the least deflected from its original path. It has the greater mass to charge ratio. So if we assume that all these ions, they have the same charge, then we can say that A is the lightest and C is the heaviest or so. Now, in this particular scenario, what is happening is A is striking the wall, A and C are striking the wall of this deflection tube. So they are kind of being neutralized and they will not be traveling towards the detector. Only the B stream, which has is kind of will be reaching to the detection chamber also. So in the detection chamber, what is happening is that your B stream that is entering, it is striking this sort of detector. It's a metal detector. So now the electron from the metal is kind of will be neutralized, will be neutralizing this ion, and the wire which is adjacent to this metal chamber, so that there is a flow of electrons in this wire. So these electrons will kind of move inside the wire to fill up the space, empty space created by that electron that has moved over to your ion. And this flow of electron is kind of detected as electric current, which can be amplified and it can be recorded. So more ions which arrive at the detector, there will be more current or so. So finally, this is the type of output that you have from mass spec. You have on the y-axis relative abundance, and on the x-axis you have mass to charge ratio. So you have this kind of spectrum there, which will indicate, which has basically separated the analytes based on the relative abundance and mass to charge ratio. There are different types of mass spectrophotometer, basically depending upon what kind of ion source, how you're ionizing your analytes, what kind of analysis method you are using. For instance, ESI, which stands for electron spray ionization, is one technique of ionizing your sample. There's MALDI. MALDI stands for matrix assisted laser desorption ionization. So that basically another kind of way by which you can ionize your sample. Then there is a lot of different types of analyzers in your mass spec. One is time of flight, the other one is quadruple ion trap, and there is FTICR, which stands for Fourier transform ion cyclotron resonance. So these are the principles is kind of different what is being used here, and some have their pros, some have their cons or so. So the technique is it is kind of developing, and it has developed quite a lot over the years. So talking about the electron spray ionization. So electron spray ionization, what you do is that you have So most of the time, this electron spray ionization technique is kind of used in combination with liquid chromatography. So you can have a sort of liquid chromatography column, which will help you to separate your protein of interest using this chromatography-based technique, and that can directly enter into your mass spec using electron spray ionization. So what is happening? You will have this thin stream of liquid. So basically, you have this capillaries, which is very less in diameter. So maybe less than 250 micrometers or so. Such a thin capillary or liquid is entering in, and that basically is kind of ionized in the process with the help of these difference in the electron potential, and this result into the formation of these ions in the electron spray ionization. In the MALDI technique, which is depicted on the right hand side, what you do is instead of having this continuous flow of liquid, what you have done is that you have co-crystallized your sample with your with a matrix, and then you provide some pulse laser onto that sample plate, so that laser is absorbed by your matrix, and this matrix kind of ionizes, and along with it ionizes your sample is also kind of ionized. So here you do not have a continuous stream of liquid flowing. In this case, you have this continuous stream of liquid flowing. So every technique or every mass spec analysis have its advantages as well as disadvantages. So ESI has an advantage that it saves time. You can have a liquid chromatography directly having sample being analyzed by mass spec. But then there is a lot of sample wastage in this technique or so. In MALDI, you need to first isolate your proteins after liquid chromatography. You isolate them, then you prepare the sample plate, and then you do it. So, but here it has an advantage that the wastage of sample is not there. So depending upon how much sample you have, what is the expected amount of protein in your sample, you can kind of choose one or the other kind of technique for mass spec analysis. There are different types of mass analyzer, as I mentioned. There are different techniques that have been developed over the time. So some basic idea I'll give you here. So one such technique is called the time of flight. So it's the simplest mass analyzer that is being used. So basically here the ions are separated based on their velocity. So theoretically, all these ions, they are formed at the same time, and they are placed at the same time in the ion analyzer. So they accelerate through this fixed potential into the time of flight drift tube. So these all ions which have the same charge, they obtain same kind of kinetic energy. So higher so the ion velocity is basically inversely proportional to the square root of mass to charge ratio. So lower the mass to charge ratio, higher will be the velocity. So this velocity is kind of being noticed here, and that is converted to mass to charge ratio. Then you have another kind of mass analyzer which are called quadruple time of flight. So quadruple time of flight usually have these 4 rods around the ion beam, and in this rods there is this dynamic electrical field. So basically this result into a radio frequency of your ion that is being noted, and this radio frequency is directly proportional to mass to charge ratio. Then you have these kind of Fourier transform ion cyclotron resonance mass spectrophotometers. So here what is happening is that the kinetic energy of your ion that is present in this mass analyzer, it's very less, and at this kinetic energy, the ions, they don't move as a beam, but they are kind of trapped in the magnetic field, and they kind of oscillates, and they have a cyclotron frequency, and that is being noted here, and that cyclotron frequency is inversely related to the mass to charge ratio. So the information is being used to measure the mass to charge ratio. And then you can have combinations of technique. For instance, in this diagram, you can see B, there is time of flight TOF1, TOF2. So it's a tandem mass spec. There are two mass specs, one after the other. Then you can have combination of different mass specs. For instance, this D, there is quadruple time of flight. There are two mass specs. This is quadruple one, the first mass spec. The other one is the time of flight. So this is also being used depending upon what you want to analyze. Sometimes some ions, they have same mass to charge ratio. So then it gets very difficult if you use only one mass spec. But when you're using two mass spec, one after the other, then this helps in the separation of those kind of ions. So this is required in those kind of conditions. So tandem mass spectrometry basically meaning that you have two mass spec one after the other, with which you can kind of get better resolution. So for instance, you have after first mass spec, you have this kind of analyte which have you are separated them based on mass to charge ratio. So you can kind of further dissociate these analyte using a number of technique like collision-induced dissociation, infrared multiphoton photodissociation, surface-induced dissociation, and then you can do a second mass spec. So basically every peak here is resolved further and will give you more information exactly what were the composition of these particular peak, and this will give you a better idea as to what was there in your analyte. Now, as I said, the mass spec is a quantitative technique. So now there are a lot of advancement in the field of mass spec. We are at a stage where we do a lot of relative quantification, or we can also do absolute quantification. Absolute quantification meaning that you can absolutely determine the abundance of your protein of interest. And relative meaning you have two samples. For instance, if you have a disease sample and if you have a healthy condition, and you can relatively say that, okay, my protein of interest is 2 times more expressed in my disease condition or not. So you can do these kind of quantification using mass spec. So nowadays they use a lot of isotopically labeled reagent in mass spec that helps in this quantification technique. These is called the label mass spec technique. So for you use these kind of sets of heavy and light reagents in your mass spec for labeling, and that help in these quantifications. So we look into some of these methods briefly. So there are some stable isotopes that are used in these mass spec techniques. So for isotopes, meaning for instance, you have carbon. Usually carbon C12 is abundantly present, but then you will have another isotope of carbon, which is the C30. That is not abundantly present. So, but you can kind of exploit this, and you can use it for labeling your amino acid, which can get incorporated in your protein of interest, and that you can kind of combine with a mass spec approach and help in quantify and do the quantitative mass spec. So you can do differential expression proteomics. For instance, you have your control, and if you have disease sample, you can kind of run 2D gels of your disease sample, of your control samples. Then you can kind of spot proteins which are present in your disease and which are not present in your control, or vice versa. And then you can digest the selected gel spot into these in proteolytic lysis. You can do a mass spec here and identify those proteins which were present in your disease conditions, which were not present in your control conditions. And then this will help you to kind of identify which proteins are overexpressed or which are not down-regulated in these two conditions. So the classical mass spec approach didn't use any kind of isotopically labeled reagent. So those are called label-free quantitative proteomics. So what do you do here is that you digest your protein into these peptide and you do this kind of mass spec analysis. So mass spec analysis, you can kind of note the peak intensity if you have, or the spectral count, which tells you about the abundance of that particular protein. You do it for the healthy condition, you do it for the disease condition, and then you can kind of compare the peak intensity of the same peptide in the two conditions or the spectral count, and this will give you a relative quantitative measurement uh, in your disease condition as compared to your healthy controls. Now talking about label quantitative proteomics, where you're using these labels, there are different ways by which you can put your label into your proteins or peptides. There are three different ways: a metabolic isotope labeling, where you can kind of provide a amino acid that has a light or the heavy label. So then your proteins will be formed either with the amino acid which has a light label or amino acid that has a heavy label. So this is at a metabolic level. Then you can do a sort of chemical reaction. So where you will have your proteins, you digest your proteins into the peptide. You can add chemically, you can add these tags or labels onto your proteins, and then you digest them into this peptide. You do mass spec with those, or you can do an enzymatic reaction to incorporate those labels into your peptide, and then you do a mass spec. So in the mass spec, what will happen? Every spectra that you have in the mass spec, now every spectra will have two, or every analyte will have two spectras, one for the light and one for the heavy one, and there will be a difference of few daltons between the light and heavy. If you are using carbon, the difference will be 6 daltons or so. So then you can kind of compare the peak intensity between the two, and you will be able to do relative quantification with this kind of method. So widely kind of newly advanced techniques in these kind of which use these kind of approaches are called SILEC or ITRAC. So SILEC stands for stable isotope labeling by amino acids in cell culture. So SILEX is a metabolic level of labeling of your peptides. ITRAC stands for isobaric tags for relative and absolute quantification. So basically it's a chemical labeling method or so. So I'll briefly go through the SILEC and these ITRAC techniques. So SILEC, as I said, stands for stable isotope labeling by amino acids in cell culture. So basically here you have for instance, this is just an example. Here what they have done, they have used a light version of lysine where carbon, where you have C12 lysine, and they have used a heavy version of lysine, which is a C13 lysine. So this is state A and this is state B. For instance, you were comparing a diseased condition or maybe a sort of kinase inhibitor, certain inhibition conditions. You want to compare those two conditions. One is A is a control condition where you use this light lysine, and B is a disease or whatever test condition where you use the heavy lysine. So then you kind of process the sample for mass spec, you digest them, prepare these peptides or so, and then you do a mass spec analysis. For every position or every analyte, you will have two spectras, one for the light one, the other one for the heavy one, which will be separated by 6 daltons or so. And then you can compare this intensity of these peaks or so, and you will be able to say relative, you will be able to do a relative quantification between the two conditions. So there are, as I said, advantages and disadvantages of SILEC. So SILEC is very precise. It allows you to do a very precise quantification because in one go you are getting the quantifications for both conditions. But it works when most of this, so this kind of work is being optimized in cell lines. But then you can work with post-translation modifications also with this kind of technique, and it is relatively inexpensive. If we talk about the disadvantages, is because there is still a limitation as to how many conditions you can do. For instance, maximum that conditions that you can kind of carry out with this kind of technique is 3. State A, B, and C also you can add actually. So that basically limits also the number of conditions that you can test with this approach. This approach is not practical for the model system. Model system, for instance, for organism, it's still not optimized. So mostly the cell lines people have used it. So this is still kind of developing. So in the meantime, another technique, iTrack, that was kind of developed, that has also overtaken SciLab. So in iTrack, we use these kind of small molecules or reporter molecules, which provide you a label. So that could be, so basically you can have a lot of different varieties of these labels. So this is named as 114, 15, 16, 17 or so. Around 8 labels have been kind of designed for this iTrack. So then you can basically have, if you are using these 4 labels, you can carry out the analysis of 4 different samples in one go. So you can basically have sample 1, you can digest it, you can label it 114. Sample 2, you can digest it and label it with 115 iTrack reagent. Then sample 3, you can label it with 116, sample 4, 117. And then you can simultaneously run them in one go into the mass spec. So every spectra that you have will have these 4 peaks. So then you can compare these 4 peaks, and this will give you information as to how sample 2 varies with respect to sample 1 and so on and so forth. So here's one example that I would wanted to discuss with you, how iTrack is useful. For instance, you have 4 conditions where first condition, you are adding a control DMSO, and you are testing a inhibitor of a particular kinase. So there are 3 inhibitors, 1, 2, and 3, indicated in this color coding in this case. Now you're labeling these 4 samples with these different iTrack labels, 114, 15, 16, and 17 simultaneously. You are enriching that sample with some phosphopeptide enrichment, so that because kinase basically phosphorylates its substrate. So you would like to enrich for the phosphopeptide. And then you do kind of mass spec analysis. So this will give you, every peak will give you, or every analyte is there, you will have 4 peaks for 114, 15, 16, and 17, indicated in this color coding here and this bar diagram. So this is the effect on one of the peptide. So you see that more or less their relative abundance is almost same, no statistically different. So you will say that in my peptide 1, there is no difference of these inhibitors. For peptide 2, for instance, you see that your 115, 17, and 16 peaks or relative abundance is very less as compared to 114, which is the control condition. So you will say that my peptide 2 is sensitive to all these inhibitors. Then if you're looking at the third peptide, at the spectra of this third peptide, you see that only in the peaks, which is in color blue and orange, which indicate the inhibitor 1 and 2, they are statistically less abundant as compared to the control. So you say the type 3 sensitive to inhibitor 1 and 3. So this gives you sort of, in one go, you get a lot of information using this kind of mass spec approach. So there is a wide variety of applications of proteomics. It is used in tumor metastasis, renal disease diagnosis, urology, cancer research, antibody profiling, study of the treatment of diseases, nutritional research, fetal and maternal medicines, diabetic research, or so. So I'm not going to details on all these applications, but here I will briefly talk about some aspects of proteomics in disease biomarkers. So in diseases, you might have heard of these biomarkers which are being identified for a particular disease or so. So what we understand by biomarkers? So biomarker is something that is objectively quantified and evaluated, and it is an indicator of the physiological process, pathological process, or the pharmacological response to a therapy. So that's a general definition of a biomarker. So these biomarkers, they can basically have, they can implicate various health and disease conditions that you have. So basically, the level and the type of exposure to some environment factor, genetic susceptibility, or markers for subclinical, clinical disease, or it could be indicator for the response of a particular therapy. There are different forms of biomarkers that one can have. For instance, it could be a hormone. For instance, in Hashimoto's diseases, TSH, which is thyroid stimulating hormone, it is used as a biomarker. The biomarker can be a metabolite. For instance, in newborns suffering from phenylketonuria, you test for phenylalanine in their urine. The biomarker can be an antibody. So basically, for instance, in rheumatoid arthritis, anti-citrullinated protein antibodies are used as a biomarker. This biomarker can be a protein or peptide. For instance, in the prostate cancers, PSA, which is prostate-specific antigen, it is used as a biomarker. Biomarker can be cell types. For instance, in infections or in cancers, white blood cells is used as a biomarker. Biomarkers can be enzymes. So for instance, in liver cancer, some hepatic enzymes are used as a biomarker. So you can have a biomarker at different levels in your disease. It can be a diagnostic biomarker, meaning that it will help you with the early detection of a disease. So you can kind of detect you have developed an understanding as to this particular biomarker is expressed more in certain individuals, suggesting that they will be they are more prone to particular disease or not. Then the biomarker can be predictive, that it can tell you that if this is how the expression of the biomarker is, or the protein expression of a particular person is, and that will be more efficacious to the drug treatment or not. It could be a prognostic biomarker, that it will tell you that what kind of treatment a particular individual will respond to. It could be a staging biomarker, meaning that it can help you to determine at what level of or what is the stage of your disease also. So you can have different biomarkers for different purposes. So I think as indicated in the last lecture about drug discovery, even biomarkers discovery also follows a long pipeline. And it takes years for you to have a biomarker which is clinically relevant. So the quantitative proteomics, basically we have this mass spec analysis with which we now have, we expect or we know how the levels of expressions of the proteins in disease conditions as with respect to the control condition. So these mass spec spectra that are being used, so they are kind of used for this biomarker discovery. So, and they basically follow these major stages in biomarker discovery, which involve first the biomarker discovery, then the validation, and the third is quantification. And then only after that, there will be clinical implementations of the biomarker. So you have, at the discovery stage, you have more than thousands of mass spec data of different proteins from different conditions or so, from tens of biospecimens or so. So one uses a sort of informatic approach in order to prioritize kind of which biomarkers you would like to take forward for validation. So the mass spec analysis, one has to be very careful, and they have to decide as to which you will consider as a biomarker, because mass spec analysis also there's a fault discovery rate of around 5% in the mass spec data, because sometimes if you have low expressing proteins or so, they are not resolved properly in your mass spec. So it depends upon your sensitivity of the mass spec that you have used, and how good or well your proteome has been collected. Then So at the but discovery stage, you usually have 10 to 100 candidate biomarkers. Then you prioritize as to which one would be the best one to go forward on your biomarker for your biomarker validations also. So you do data mining at that stage, basically. So you have tens to hundreds of proteins. You try to understand as to what is the tissue of origin, does that match the kind of disease uh, that a person has developed. You do pathway analysis. You try to understand as to what is the pathway in which these biomarkers play important role. Does that make sense? So a lot of bioinformatic work is kind of part of proteomics in order to biomarker to come up with a quantify a final clinically relevant biomarker. You also do a lot of literature searches, available immunoassays, multiple reaction monitoring. So at that, by this stage, you have approximately around 10 verified biomarkers. Then you go forward towards the quantifications, where you kind of they take these 10 proteins or so from more samples, like thousands of samples, and then you have to do a sort of immunoassay. For instance, ELISAs or protein microarrays are widely used at that stage, where you will try to validate that particular or quantify that particular biomarker, and you will be able to see exactly how it is behaving differently from the disease from the control conditions. And this is how finally you verify it, and then this is something that will go forward towards clinical implementations also. Now, proteomics has also quite developed in the field of food technology. So a lot of work we will find in the field of proteomics, where it's been implemented for the food safety. You try to look into the allergens which are in the food, the microbial proteomics, which is required for developing of your crops, and nutritional proteomics also, where you look into the gut microbiota, nutrition health, digestomics. Then food quality, where you try to look into the foods which are rich in protein, how the properties of your food is affected by some processes or modifications also. So just to summarize the proteomics technologies here. So there are different types of techniques which are kind of developing in proteomics. They work together in order to gain an overall idea about the proteome in the living system. So you have conventional techniques like chromatography, ELISA, Western blotting. Then this is followed by advanced technique, which are gel-based approaches, mass spec, protein microarrays. Then you have quantitative techniques. These are the SILAC, ITRAC techniques that we discussed. Then other than that, which we didn't discuss it, is also some high throughput techniques like X-ray and NMR, which give you information about the structure of those proteins. And this is combined with bioinformatic analysis also. So I'm not touching upon the bioinformatic analysis, but that is a very important part of proteomics. So I'll skip there. So there are actually a number of databases which you will find, which will help you to do bioinformatic analysis on your protein sequence, about your protein structures, motifs, or so. So here is a list. So you have a lot of databases where you will find your protein sequences. Then you can basically look into the phylogenetic profiling based on your protein homologous regions in your protein. You can look into the protein domain servers. You can basically identify motifs on your proteins. Structural databases are also there. You can do modeling, protein modeling also. So that will, these techniques or these tools are very important tools to do predictive structure-based proteomics work also. So I think these are some of the references that you can see for mass spec. There are certain papers which are also you will find on the internet. So these are very important resources. You can go forward and you can read more about the mass spec analysis. So I think with this, I'll stop here. So thank you for your time.
Thank you so much, Dr. Priyanka, for a very wonderful session. So I would now request participants to ask questions. If there are any questions, you may directly write in the chat box, or you can raise your hand to interact with Dr. Priyanka. I see there are 2-3 hands that have been raised. So maybe Rajesh, Anand, maybe you can ask, followed by Dr. Surajit.
Yes, hello.
Hello, yes.
Ma'am, good morning. Thanks for your wonderful presentation. I have some couple of questions. Actually, I'm planning to work in some tumor sample, human sample. And what we had is that it's a different experience. Like some patients they have less expressed proteins, and some patients have very high expression of proteins. How to manage those things? Because I have to analyze the samples in different time scale, not all in one time. So I have to wait for the sample from one time. Maybe after a couple of weeks, I may get other sample. So how can I manage if the patient's the tissue biopsy comes with less expressed proteins?
So basically, you're saying that you have tumor samples and you have protein expressed in some tumors, and that varies with time.
Yes, no, actually,
or age.
yes, no, actually, I'm planning for differential protein expression analysis.
Okay.
I guess some tissue biopsy comes with a less expressed proteins, so I like to make it uniform. I don't know how to do this. I'm just
Basically, you're using for this tissue on this
Well,
what kind of process?
Actually, I'm planning to outsource the sampling for label-free tissue CMS.
So mass spec technique you are saying, right?
Yes, ma'am. Yes, ma'am.
So I think that will you have to see. Basically, it depends upon the number of samples. Sometimes you will find that a lot of quantified mass spec data is available. So you have to basically also validate as to so at the end the protein is going to be a biomarker for your disease. So you have to so more the number of sample basically that is better. So so this is something you have to basically have right controls. So if you have a sort of age dependent variations, then you have to restrict yourself to a particular age, or you should have a control for that particular age or so. And then you can also go on to these open source some mass spec work be there. So then you can kind of compare it to that biomarkers, and then you have to probably take help of some bioinformatic approach also, wherein you can try to have an understanding that the protein that you've identified, it makes kind of sense that in this particular disease it is kind of misregulated or so.
Okay.
So that is why I mean proteomics is quite challenging. This is a reason because it's very dynamic. It varies also from individual to individual. So you should have a control. Right now, the problem is that when you people come with a disease to you, then the right control would be the same person, maybe without disease and with disease, which is not practically possible, basically.
Yeah. Actually, I'm thinking the
So
same.
then you need to, you need to have a sort of window, or the patient, or the sample that you're collecting. You have to define a sort of age, gender, maybe certain factors you have to keep into consideration, which can influence your proteomics. So you should have a proper control group for that and the disease group. That's the only way. And maybe take help from some other available tool of information which is present in this respect.
Actually, I'm planning to send, like you're saying, I'm planning to include replicates, like because some samples so that I can get some idea. The thing is, all the label-free MS techniques are really comes with expensive. So among
proteomics, there's a challenge actually. That is why I'm saying that like if you compare proteomics to genomics is more robust.
Yeah.
It's about the sequence in your sample, but proteomics is more dynamic. So it will be challenging, but I think this is something that will work out. More samples you'll collect. If it's a more exhaustive study, more number of patients, then they will give you more reliable data. Otherwise, you rely on some diagnostic tools also only. Like if you already know certain biomarkers from a virtual screen, virtual biomarker work, then you can kind of just check for the or prepare a sort of diagnostic method for that particular protein of interest and see if that is overexpressed in your tumors or so.
Yeah. And one more thing, Sam. In your experience, could you suggest me any techniques that have high proteome coverage but with a less cost? Because I'm planning to include nearly 15 samples. And if I'm planning for all the sample outsourcing, it's cost a lot of money because I'm not sure the fund all goes for only for screening. So this is only one side of the.
So basically, you're asking me that a sort of.
Could you suggest me any techniques other than label free, which having higher protein coverage, but also comes with a less cost? Something?
So as I said, mass spec will give you a
yes,
total
mass spec. Yes, yeah, MSMS.
mass spec, and that you're saying you are outsourcing. But if you want, you can kind of also, if you already know, for instance, that there is a certain biomarker which is overexpressed in tumor, sometimes this kind of work is already being done. Then you can kind of develop some ELISA-based technique, which can be done in a proper small scale in a sort of a molecular biology setup. You can do that. You can do a sort of ELISA or maybe a Western blotting technique or so, where you just need a solubilized soluble protein, and then you just run them onto STS page. You do a Western blot, and you see a protein is expressed. So if it has in control it is not expressed, and in this it is expressed, then these variations you can very easily see by Western blotting. That's the most simplest thing one can do in the lab. So Western blotting or some ELISA based technique. ELISA is also very simple. It requires some skill set to learn, of course, but that is something which is doable actually.
Okay, there's just one last more question. So in your slide, I saw that we are getting the samples, then digesting, and then we're going for 2D gel, and then we're going for MSMS. For label 3, is it necessary, mandatory 2D gel route, or we can go directly digesting the sample, then we can go directly for label 3 MS? What is the best way to do this?
I think 2D gel is now not necessary. As I said, now we have this sort of shotgun proteomics approach where you can just prepare your sample, solubilize your proteins, and you can send it directly for mass spec. It can be digested in solution. You don't need to run on a gel. But if you, so then you are basically working with the entire proteome, which is there in the soluble protein. But if you're running a sample, you're cutting a specific band, then you're already pinpointing that I'm interested in that particular protein or so. And if a protein is visible on a gel, then it is also highly expressed protein. Then, of course, you have much cleaner mass spec for that.
Okay.
Yes, so there will be cleaner mass spec. So basically, you should be aware of what kind of protein you're looking into, what is the expression profile of that protein. Well, I mean, how much expression it could be in that particular cell type. So this kind of information also you can get on a lot of these bioinformatic databases are there. So they tell you the expression profiling of these proteins in different tissues, cell types or so.
Okay, ma'am. Thank you so much. Thank you.
Okay, thank you.
Thank you, Dr. Priyanka. Maybe I can, Dr. Surajit, you want to ask next? Questions in the chat box, you can write. Okay, so it seems there are questions. Thank you, Dr. Priyanka. So before you leave, I would like to request all the panelists to turn on their video so that we can take one photograph if possible. And also, if someone from attendees wants to join for the photo, they can also raise their hand so we can promote them to in the panelist platform. Ashish, can you check if somebody from the attendees wants to join? for the photo.
Here no one is displaying.
Okay, so Radhir, maybe you can turn on your video there. Fine. So we can take also we get the opportunity to take the photos in the last session. So I think that is fine. So thank you once again, Dr. Priyanka, for your valuable time. And this we would like to take a pause, and we will be resuming at 3 p.m. again. So we will have a valedictory feedback and test session at 3 p.m. So I request all the participants to join sharp at 3 p.m. because we will be having a test also, and you will be getting a digital certificate based on your performance in the test. So please be there at 3 p.m. And with this, we would like to pause. Thank you, and then see you at 3 p.m. Afternoon, everyone. So I'm posting the link for the test in the chat box. I hope you will get it, everyone. So please note that there are 25 questions of MCQ types, and you are supposed to finish in 30 minutes. So we will be receiving the responses by 3:35. So please ensure that you submit in time, and good luck. Dear Ram, I hope you were able to finish your test. So we are going to close in next 1 minute. So please submit your responses. So dear participants, so we will be sharing now the link for getting your feedbacks for this 5 days of workshop. So the link is shared now in WhatsApp group also that we created, and also it will be shared here in a minute. So it is there now. So please fill this feedback form. Take your time until 4 p.m. Then we will be resuming at 4 p.m. to start with the closing ceremony. So let's start. So good evening all, and we are almost at the end of the workshop, 5 days workshop that we had, and this is feedback and testing session that we have already performed. So I hope everybody submitted the feedback and also submitted their quiz results. So let me formally begin with the closing ceremony. So I'm very thankful to Atal Academy for sponsoring this interesting and useful FDP program on multi-omics data science that was held from December 20th to 24th, 2021. This workshop FDP program was organized by Department of Bioscience and Bioengineering at IIT Jodhpur. So I have been coordinating this interesting workshop, and I had a lot of exciting things to do, and also learned a lot of things during the course of this workshop. So myself, Pankaj Yadav, I am assistant professor at Bioscience and Bioengineering Department, and this is my academic record. I have obtained my PhD in statistical genetics from University of Kiel, Germany. And prior to that, I obtained my MS in life science informatics from University of Bonn. And also my first degree was in Bachelor's of Biotechnology from Punjab University. So let me just thank all the speakers first, all the resource persons who kindly agreed and also spend their valuable time to give insights and to help the learning and transfer of knowledge to all the participants. So in day one, we started with the inauguration ceremony by Professor Santu Chaudhary, who is the director of IIT Jodhpur. And then we started off with our first formal session by Professor Guneet that was titled as Yoga is Way of Life. And we learned many things, and also during this session we also learned how to meditate, how to keep away stress and all. So this was very useful session by Professor Guneet, and this was followed by a very interesting session by Professor Mitali Mukherjee, who is also head of the department at Department of Bioscience and Bioengineering at IIT Jodhpur. She gave a talk on connecting science with the people, how different genetic variations are associated with different phenotypes, how we can connect genomics with the phenomics, and what are different tools and a lot of other interesting findings that she has got from her rich experience that was shared by Professor Mitali with us. The session 3 was organized by Professor GPS Raghava, who is professor at IIIT Delhi. He has been a father of bioinformatics in India, and I have been honored to also take some lectures during my bachelor's course from him. He gave very interesting insights in cancer treatment using multi-omics approaches, and also he gave very basics and fundamental understanding of various concepts in multi-omics and various tools and techniques, and also about different databases that he has in-house developed, and also he gave us very good insights to pursue our career in this domain. So on day 2, we had privilege to have Professor Suresh Kumar Sharma as our first speaker who talked about genetic association studies, and also he gave very fundamental understanding of various statistical concepts that are used day to day in life in bioinformatics and data science. Later in the second session, we learned about basics of deep learning by Dr. Alisha Praveen, who is from University of Rostock University in Germany. She gave a very good understanding of basic concepts of deep learning, where we can find the applications of deep learning in biology, what are different tools and techniques that are available for use. On day 2, in the third session, we had Dr. Avinash Sharma and CCS Pune. He gave a very good understanding of microbial genomics. He gave different examples and case study that he has used in his work to explain the concepts very well. So I'm thankful to all these speakers. And on day 3, the first session was organized by Dr. Prashant Shobajala from Amrita University. He gave very interesting and interactive session on introduction to systems genomics, where he also discussed very interesting case studies that he has performed. And also he gave insights how we can use the systems genomics in solving different biological problems. Session 2 was taken by Mr. Arpit from Sophie Bangalore. He gave some hands-on prediction of breast cancer tools. Also, he is an expert speaker in artificial intelligence and data science. Almost every week he is organizing sessions, and it was also a great privilege to have him here for our workshop. He has given a rich experience in hands-on training, in prediction and application of machine learning and other approaches for prediction of diseases using open source data sets. Later we had in session 3, Professor Muthu Raman, who is assistant professor at University of Mainz in Germany. He has a rich experience in research, and he talked about in his session about translational models in neurology. He has taught us what are different concepts in neurology, from basics to different transdisciplinary aspects of neurology, where we can define mathematical formulations of translational models, how we can develop, how we can design, how should we approach in designing these kind of translational models in neurology. So I'm also thankful to these 3 speakers, day 3. Then on day 4, we had the opportunity to have Dr. Abhishek Kumar, who talked about introduction to oncogenomics. He explained various pipelines that he has developed during his work, also gave very basics and important understanding of the use of genomics in cancer research. Then second session was organized by Dr. Ravi Shankar, who is an expert in microRNAs, and he has been working as a principal scientist at IHBT Palampur. He gave very interesting and interactive session on ruminating the microRNAs, how microRNAs are interacting with our genomes, how they have played an important role in etiology of the different disease. So it was a great pleasure to have him here. Next, the day 4 session 3 was organized by Dr. Dilpreet Kaur, who is assistant professor at IIT Jodhpur. She gave a talk on matrix methods in data science, which is a very fundamental concepts in matrices and their application in day-to-day life of data science. So these are very core concepts that she has gave us, and it was very useful in practice for all the participants. Hopefully they will be able to use all those concepts and develop a better understanding of these approaches that are used in data science. So I'm also thankful to these speakers. Then on day 5, that is today, 24th of December 2021, we had the opportunity to learn very interesting concepts from Dr. Sandeep Murarka, who gave us an interesting talk session on hit to lead in drug discovery. And it was very much clear because he explained all the concepts with the help of case studies with real life examples. And he also explained very well the procedure and the pipelines that are used in drug discovery by pharmaceutical companies and also in research organizations. Last session of the workshop was organized by Dr. Priyanka Singh, who is assistant professor at IIT Jodhpur. She gave a talk on tools and techniques in proteomics, and she gave a very basic and fundamental understanding of these proteomics tools and techniques that are very much useful in multi-omics data science. And with this, I would like to also thank both the speakers for the today. And now we are in the final session of this workshop, that is the closing ceremony session. Besides this, I would like to, before I stop, I would like to thank all the people who are involved somehow in helping out, conducting, organizing this workshop. So FDP conveners, Dr. Dilpreet Kaur and Dr. Priyanka Singh, I'm very thankful to both of them. Then I'm also thankful to Professor Santanu Chaudhary for providing this platform at IIT Jodhpur to host this very relevant workshop. I'm also thankful to Professor Mitali Mukherjee for addressing and the inaugural session and telling the participants about different initiatives of the department, and also for delivering a talk on genomics and phenomics. So I'm also thankful to all the students who have provided a backend support, Mr. Rajveer, Mr. Ashish Kumar, Mr. Sunil, for providing all the technical support, Mr. Roshan, Ms. Vedika Dhiman, and also I'm thankful to the whole IIT Jodhpur community for providing this platform. Not the least, I'm also thankful to Atal Academy, who has sponsored, provided financial support to organize, to host this workshop. And to all the participants who eagerly attended attentively all the sessions of this workshop. So with this, I would like to stop here, and if there are any thing that participants would like to share with us. Before we stop, we will also be request some participants to join us to take the photographs so that we can remember all of you that you were here, and hope you have learned and had a good time during these 5 days. So participants or anyone from panelists who wants to say something, please feel free to raise your hand. So maybe some of you who would like to take the photograph with us, please feel free to join us. Please raise your hand so we can shift you to the panelist, and then please turn on your videos so that we can grab some pictures. Please also check if there are any resource person or speakers who are in the attendees. Please promote them to panelist.
Okay, sir.
So I request to please turn on your video for 2-3 minutes so we can grab some pictures. Okay, so I'm going to now take the snapshots. So please smile. Okay, maybe one more. Thank you. Thank you very much. So you may turn off your videos if you want. Thank you so much. And with this, I would like to now announce the closing of this workshop. So please feel free to write us in case of any queries, or if you would like to interact with any of the resource persons, feel free to reach out. We have already created WhatsApp group. We will keep this WhatsApp group for 1 week or so that you can put any queries if you have. Thank you.